Assalamualaikum buat semuanya. Assalamualaikum. Selamat siang semuanya. Mohon maaf Bapak dan Ibu. Ini saya yang goyang karena saya berada di jalan ini.
Oh, enggak Bu.
Ya, saya jam 1 ini soalnya ada acara,
bisa,
jadi harus berangkat.
Bisa hadir.
Kan semua ya.
Ya, Dokter Rahman.
Think of
Halo, Dokter Saif.
You
call me ya, Dokter Saif. Saif, you call me.
Ya Bu, selamat
I'm so sorry ya.
pagi, selamat
I'm sorry.
sore.
I don't take your phone because I'm in the I in the car now.
Jo.
Nah, kalau lurus nggak apa-apa.
Oh iya. Your background look like this.
Dr. Saif, I think your connection is unstable because we could hear your voice is para narasumber sepertinya belum join ya Ibu. Tapi para narasumber sepertinya belum join ya Bu Yola ya.
Kontak dulu ya.
Iya. Hati-hati Prof. Mirni di perjalanannya sambil disambi.
In the 1 o'clock p.m. I have the appointment, so I don't remember that the caster apa namanya on the 12:30 ya.
Yeah. Yeah, 12:30. Yes.
12:30, yeah. So I must go. I think it doesn't matter lah. Apa-apa, it's okay lah. Rahan, you still in Kediri now or in Surabaya?
Yes, I'm in Kediri now. Yes.
It's very nice. Yeah, nice city. It's very busy city, I think.
Yes. Very nice city, Prof. It's not really crowded. Yeah.
City,
Ini kayaknya jaringannya di sini nggak bagus nih ya. Kok suaranya menggema ya? Ini jaringannya di sini nggak bagus nih ya. Kok suaranya menggema ya? Prof. Aini, welcome to our event. I think Prof. Aini have joined with us now.
Yeah, thank you.
Assalamualaikum Prof. Aini, how are you?
Waalaikumsalam. Waalaikumsalam.
Assalamualaikum Prof. Aini.
Ya, apa kabar?
Sehat, Prof.
Alhamdulillah
Alhamdulillah.
sehat.
Alhamdulillah.
Assalamualaikum Prof Aini.
Salam. Alhamdulillah dapat kita ketemu hari ini.
Alhamdulillah.
Walaupun jauh.
Walaupun
Ya,
jauh.
walaupun jauh. Ya.
Jauh di mata tapi dekat di hati, Prof Aini.
Insyaallah.
Tanya Ibu sekarang sudah jadi dekan fakulti.
Iya, ya, I
Congratulations.
represent
Ya.
Profesor Dr. Puji Srianto on September 2020.
Yes, yes. Oh, baru ni. Okay. Tanya.
But you still looks beautiful, Prof Aini.
Oh. Thank you. Saya sudah habis term sebagai Night Chancellor.
Bahasa Indonesia cantik.
Yes.
Students ramai ya.
Yeah. You know the Professor Fatima? You know her?
Fatimah
Professor
mana?
Fatima. Fatima in aquaculture.
Fatimah apa?
Profesor
Oh,
Fatima.
tahu.
Tahu.
Ya, Fatimah Yusuf.
Yeah. You know her.
Yeah. Very well. She's my relative.
Very well. Yeah. I go to UPM in 2019. Last year I go to UPM.
Last year.
Yeah, because last year I attended in the Faculty of Fisheries and Marine. Before I attended the faculty, 2015 until 2020, I became the dean of the Faculty of Fisheries. And now I come back to veterinary medicine. I come back to our faculty.
Faculty. Oh.
Yeah.
Balik kepada asal ya.
Yeah, I am the same department with the Professor Romsia. I am the second to him in the department.
Yeah.
Prof Romsia is he have the expert in the nutrition. Same with me.
Saya ke sana tak jumpa ya Ibu.
Oh, saya tahu Ibu, saya tahu Prof Aini, so I know you. I think it is not different. Beberapa tahun yang lalu, you still beautiful.
Semuanya beautiful, semuanya beautiful. Yeah.
Prof Zunita haven't joined us, I think, Prof Yola.
Prof Zunita belum masuk ya?
Iya, belum masuk lagi, Prof.
Agaknya dia. Nanti saya message dia.
Mohon maaf, saya izin untuk memberikan pengumuman sebentar ke mahasiswa, boleh, J?
Monggo, Dokter Aan biar segera dimulai.
Iya.
Bacaan dimulakan dulu aja.
Iya, oke. Prof Willy. Baik, sebelum acara dimulai, saya memberikan sedikit pengumuman untuk mahasiswa, khususnya untuk semester 3. Tugas ringkasan tersebut harus diserahkan dilaksanakan sebelum tanggal 21 Desember. Dan masing-masing komite untuk mengumpulkan secara kolektif begitu ya dan diserahkan kepada PJMK masing-masing mata kuliah. Ya. Jadi submit the assignments to the subject coordinator. Terima kasih. Ini bisa kita mulai dulu atau menunggu Prof. Zunita, Dr. Yola, atau Prof. Mirni, bagaimana Prof? Sebaiknya.
Menurut saya nanti kita mulai saja, tinggal nanti sambil
Begitu ya?
Nanti biar tidak terlalu. Atau mau ditunggu 5 menit lagi? 5 minutes again.
Yeah.
We waiting for her 5 or 10 minutes. It is okay.
Karena kuliah pertama. Oke, Prof.
Ini. Coba ya, tengok kenapa. Semua yang kat situ dah tak nak buka. Dan semua dah tak buka.
Sebab dia yang ni sangkut kat sini. Dia tutup tu.
Okay, to all the participants, our event is about to begin. So please kindly remain online and thank you very much for preparing yourself to join our event. Assalamualaikum warahmatullahi wabarakatuh. A very good afternoon to all the participants. The Honorable Dean of Faculty of Veterinary Medicine, Universitas AIRLANGGA, Profesor Dr. Mirni Lamif, Doctor of Veterinary Medicine, Master of Agriculture. The Honorable our guest lecturer, Profesor Datin Paduka Dr. Aini Idris from University Putra Malaysia. The Honorable Professor Dr. Zunita Zakaria from University Putra Malaysia. The Honorable all lecturers and all the participants of this event. First of all, let us pray and praise to Allah Subhanahu wa Ta'ala. Because of His bless and mercy, we can come together without any obstacle with healthy condition. I'm very pleased to see you here, and it is our pleasure to welcome you to our guest lecture event, proudly organized by Bacteriology Department, Faculty of Veterinary Medicine, Universitas AIRLANGGA. This guest lecture event is a routine program as a part of the Virology and Bacteriology course for third semester students. And for today, the lecture theme is Molecular Virology and Bacteriology, which invited two guest lecturers from UPM, Malaysia. So before we start the event, I would like to share the agenda of this event. We have several agendas are as follows. The first agenda is opening. The second agenda is opening remark by Head of Microbiology Department, Dr. Yola Rahmathani, Doctor of Veterinary Medicine, Master of Health. The third agenda is opening speech from Dean of Faculty of Veterinary Medicine, Universitas AIRLANGGA, Professor Dr. Mirni Lamid, Doctor of Veterinary Medicine, Master of Agriculture. And the fourth agenda is lectures from the guest lecturers. The first lecture will be delivered by Professor Dr. Zunita Zakaria, and the second lectures will be delivered by Professor Datin Paduka, Dr. Aini Idris. And the fifth agenda is question and answer and discussion session. And the sixth agenda is certificate awarding. The seventh agenda is closing remark by Secretary of Microbiology Department, Dr. Wiwi Yasningsi, Doctor of Veterinary Medicine, Master of Health. And the last agenda is closing. Okay, the first agenda is opening. Let's start this agenda by praying according to the respective religion and belief. Let's pray together first. Pray begin. Pray finish. Thank you very much. So the second agenda is opening remark that will be delivered by Dr. Yola Rahmathani, Doctor of Veterinary Medicine, Master of Health, as a head of microbiology department. To Dr. Yola, the time is yours.
Okay, thank you, Dr. Aan. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Very good afternoon to all the participants. The Honorable Deans of Faculty of Veterinary Medicine, Universitas AIRLANGGA, Professor Dr. Mirni Lamid, DVM, Master of Agriculture, the Honorable all speakers, Professor Datin Paduka, Dr. Aini Idris, and Professor Dr. Zunita Zakaria, the Honorable lecturers of Microbiology Department, the Honorable all students and all participants. Welcome to everyone. First of all, let us pray and praise to the Allah Subhanahu wa Ta'ala, who has given us health and opportunity so that we can attend today's guest lecture event. On behalf to the committee of the guest lecture event, I would like to thank to all of you for joining today's event. This guest lecture event is a routine program as a part of the course of Virology and Bacteriology lecture for third semester students. Therefore, this event was attended by microbiology lecturer and third semester of undergraduate student. And for today's guest lecture, the theme is molecular virology and bacteriology, which invite two guest lecturers from University Putra, Malaysia. Through this event, we hope we can get and enhance our knowledge from the speakers. We would like to take this opportunity to express our thanks to our special guest, Professor Datin Paduka Dr. Aini Idris and Professor Dr. Zunita Zakaria from University Putra, Malaysia for accepting our invitation and sharing your valuable time and sharing your knowledge. Once again, welcome to the event, and finally, we hope that all of you would follow the program by pledge, and program will be beneficial for all of us. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Yola, for your opening remark. So, for the next agenda, opening speech from Dean of Faculty of Veterinary Medicine, Universitas AIRLANGGA, Professor Dr. Mirni Lami, Doctor of Veterinary Medicine, Master of Agriculture. So Professor Dr. Mirni Lami, the time is yours.
Thank you, Dr. Aan. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon, ladies and gentlemen, and also our student. See you again in the guest lecture. Welcome to International Guest Lecture, Faculty of Veterinary Medicine, Universitas AIRLANGGA. Thank you very much for your presence. It is a very good to see all of you. And let me start by wishing you all a happy and healthy. First of all, I will introduce myself. I'm Professor Dr. Mirni Lami, DVM, Master Agriculture, as the Dean Faculty of Veterinary Medicine, Universitas AIRLANGGA. I would like to thank you, all our speakers, Professor Paduka Dr. Aini Idris and Professor Dr. Sunita Zakaria to attend lecture in this event. Greeting to Professor Paduka Dr. Aini Idris and Professor Dr. Sunita Zakaria, because I'm the replacing Professor Dr. Puji Srianto. University Putra Malaysia and Universitas AIRLANGGA have a long history in collaboration education and research. I hope that it can be improved in the future. International guest lecture, I think it's important to improve our academic member knowledge, and we can sharing our experience with our guest lecturer. I would like to thank all participants who have joined this guest lecture. I hope this event will be successful and we can gain our knowledge. I hope we will be able to meet all speaker and participant in the next event. Especially thank you for the committee, Dr. Yola and Dr. Wiwi as head of the divisi microbiology, Dr. Aan, and all our students and all the participants. Thank you for all. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon, all of you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Prof. Mirni, for the opening speech. So the next agenda is the main agenda, which we are waiting for, lectures from the guest lectures. But I think the first lecture will be delivered by Professor Dr. Sunita Zakaria. But I think Professor Sunita hasn't joined with us right now. So Professor Aini, would you mind if you are presenting now? While waiting for Professor Sunita.
I'm already here, Dr. Aan.
Oh, I'm sorry, Professor Sunita. Sorry, I didn't know that you have joined. Hello, Professor Sunita.
Hello, how are you?
Yeah, I'm sorry, I didn't know that you have joined with us. Thank you for accepting our invitation and become our speaker today. So, are you ready to present?
Yes,
now?
I'm all set.
Prof? Okay, Prof. Thank you very much. So, okay, for the next agenda is a presentation from our first guest lecturer, Professor Dr. Sunita. With Professor Dr. Sunita, sorry, may I read your CV first, briefly? May I, Prof?
Yes, yes.
Yeah,
silakan.
Okay. Terima kasih, Professor Zunita. Okay, so Professor Zunita Zakaria, currently she is a professor at the Department of Veterinary Pathology and Microbiology, Faculty of Veterinary Medicine, University Putra Malaysia, and also Director, Institute of Bioscience, University Putra Malaysia. And for educational background and experience, Professor Zunita graduated with Bachelor of Science in Microbiology from University of Malaya, and she pursued her postgraduate study at the University Putra Malaysia, receiving Master of Science degree in Veterinary Microbiology, and obtained her PhD degree in Molecular Biology in 2002 from University Putra Malaysia. Her research area are Bacteriology, Molecular Biology, and International Biotechnology. She's also actively produced many international scientific journals and proceedings in a related field, and she's also actively attending the scientific congress as an invited speaker and a speaker as well. So that's the CV from Professor Dr. Zunita. Please welcome to Professor Zunita. The time is yours, and you have 90 minutes presentation time. Thank you, Prof.
Thank you very much, Dr. Aan. Assalamualaikum and a very good afternoon to everyone. Actually, I feel very happy, very privileged, and I feel very honored being able to be present today to share, some experiences with all the audience today. First, I thank the organizer led by Dr. Yola, and of course, other VIPs who are present today, the Dean, and the others. And I can see here there are over nearly 300 participants, right? So that's why I feel very, very privileged, to be able to be present today. Okay, Dr. An, thank you very much for your kind introduction. So you have given me 90 minutes, so I'm not sure whether I can fulfill that 90 minutes, but I'll go slow and steady. So if let's say if my accent is not that clear, perhaps you can stop me. Yeah.
Okay,
so I have been given the task to cover molecular bacteriology. Okay, so that is a very, very big topic. Yeah, and then from what I know from Dr. Yola, the participants today will be those are in your third semester, meaning second year of DVM, right? And then today I got to know that there are also microbiology students. So, in order to give you some introduction on molecular bacteriology, so I've shortlisted a few learning outcomes. So hopefully by the end of this 1 hour or so, you'll be able to explain the basic molecular techniques in bacteriology. So because the area is so vast, so I've picked a few that we'll go through this afternoon. And then the next one is to explain the application of polymerase chain reaction, PCR, in veterinary diagnostics. Yeah. And then the last one would be to differentiate the conventional and molecular techniques in bacteria identification. So those are the 3 learning outcomes that I hope that we can achieve by end of the lecture session. Okay, so let us go through. Okay, so being in the veterinary faculty, yeah, so I'm sure you'll have a section where you do diagnosis, right? So diagnostic tests are the essential tools for confirming the health status of animals. And at the bacteriology lab, normally you'll receive samples and you will carry out and to identify the pathogen. In the sample, if there are pathogens, so in bacteriology lab, you carry out some test in order to identify what pathogens there are. Yeah, and failing or delay in pathogen identification may be fatal to your patients. Okay, so it's very important that you will have a very, very efficient method how you can identify pathogens and treat your patients. Right?
Sorry, Professor Zunita, sorry for
yes?
interrupting you, but I think you haven't changed your slide presentations.
Is it moving now?
No, it's not moving yet.
Oh, okay.
Yeah.
It's moving in.
No, still we only see the the picture of you and the.
Let me try again.
Yes.
So is that?
Yes. Now we can see.
Is it moving?
Yes.
Okay. Oh dear. So for the learning outcome side.
Sorry, Prof.
It's okay. So so just now I was explaining
we can see
about the
slide
learning outcomes.
Okay.
Okay, but then there is some interference of sound, so I'm not sure. Are you hearing me clearly?
Yes, we can hear you clearly, and also we can see the
Because here
screen as well.
yeah, because on my side I can hear a lot of interference, but anyway, so these are the learning outcomes. Just now you were not seeing that just now, yeah. So it's to explain the basic molecular techniques in bacteriology, explain the application of PCR in veterinary diagnostics, and also to differentiate the conventional and molecular techniques in bacterial identification. Okay, so this slide I have explained just now. So I stressed the importance of us, in the veterinary field to be able to identify the pathogen very, very efficiently. Because if we don't have those, it will be very detrimental to the patients. Yeah, and if the patient is sick, of course we want to treat the animals. Okay, so what does encompass the ideal diagnostic method? So of course, the first one, we want it to be sensitive and specific. Okay, that is number one. The second one, of course, in any labs, we want it to be rapid. We don't want, something which you take like 2 months, to identify the bacteria. We want something which is fast. Yeah. And of course, anybody would want something which is easily to be performed. Okay, so rapid method, you can have the results within a day or two. So we want something like that. And it is widely available. Okay, so I'm sure being a DVM student, you have also some knowledge on what are the current methods being used. So we don't want something which is very high end. We don't want something which you need to go for sequencing of genomes or whatnot in order to identify a pathogen. So it has to be widely available, whereby any simple labs will be able to carry out the test. And the last and but not least is the cost effective. We want something which is quite cheap, right? Okay, so I'm going through now the conventional method. Okay, so by year 2, I would assume that you have carried out some of these basic methods, whereby, we have specimens. If you have a sick animal or a farm having problem or whatnot, diarrhea, and then you collect fecal samples. Okay, so once you reach to the lab, the first thing that you'll do the direct smear, right? Direct smear, you have a slide. So you'll directly smear the sample onto the slide, and then you'll be able to see what you should be culturing, in a media. Yeah, and then you should also culture on appropriate media. When I say appropriate media, so you would already know what you want to isolate. For instance, if the symptoms are, showing you is salmonellosis, so you should be able to use selective media like XLD, yeah, lysine deoxycholate or brilliant green agar and whatnot. Yeah, if you don't have any idea, some of your clients might be sending you samples saying, okay, isolation and identification of bacteria. So what you should, the media that you should use should be a very, very common media, but it's rich. So you use blood agar, right? And then from there, you'll do the isolation. And then from the colonies growing on the media, you'll perform series of biochemical tests. So perhaps you have gone through some of the biochemical tests. I'm sure, if I ask you will be able to say, triple sugar iron, oxidase test, catalase test, sugar test, the list go on and on and on. Okay. And then from there, you gather all the results, and then you'll be able to identify the bacteria. How long does it take to do all those things? Yeah. If you do biochemical tests only 1 or 2, then perhaps it's like 4 days. But if you end up doing, a series of biochemical tests, most probably you end up, the list will be 6 days. Yeah. And once you have the bacteria, you would want also to carry out antibiotic susceptibility test, because, you know, if it is caused by bacteria, you want to treat your patient, and to have an effective antibiotic to treat your patient, you would have to have the results from the antibiotic susceptibility test. So you add in another 2 days. Okay, so that is conventional. Yeah, and this slide shows the selective media that I said just now. So by using selective media, most of the time you can reduce the time of identification maybe by 1 or 2 days, and it is more specific. Yeah, and nowadays, you know, if your lab, you know, is able to spend you know, a lot on diagnosis, most probably, you know, you can invest and buy this chromogenic media, where they'll give different colors for different kinds of organism. Okay, so you can have an idea now. Okay, you do conventional tests, but at the same time, you'll have all these new products coming out, which will also help you how to do the identification. Yeah, and apart from that, you can also use identification kits, you know, rather than doing a lot of biochemical tests. Okay, so if you see here on this slide, let me just annotate and have some, yeah, mouse. Can you see my mouse now? It's moving. Dr. Ann, can you see my mouse?
Yes, yes, you can see, Prof.
Okay. Okay, so here you can see, you know, a list of different biochemical tests. So rather than doing tubes of glucose, fructose, maltose, or if you see here, you know, all the different sugars here. So it saves you time. So once if you have a pure culture of the bacteria, you can just, you know, load it in this kit and incubate, and then the next day you come and read the results and put it in the database. In the end, you'll get, okay, 90% probable, it is probable E. coli, 85% is Pseudomonas aeruginosa. Okay, so there are many conventional methods that you can use. Now it's not, my slide is not moving. Okay. Okay, so let us go again here. So this is isolation identification of methicillin-resistant Staph aureus. This is just to give you an overview of the flow. Okay, so you have a sample here. Okay, and then you put it in the buffer peptone water, and then you culture on blood agar. So once you have the culture the next day, yeah, so this is you subculture the next day, and then you use selective media. So this is mannitol salt agar. So this is a selective and differential media. Yeah, and then from here you do agglutination test in order for you to differentiate whether it's Staph aureus or non-Staph aureus. Yeah, and then in order for us to differentiate whether it is resistant to methicillin or not, we use another selective media. Yeah, and once it is blue in color, if it turns out in blue in color, then you say, okay, in the end, this sample that you have in the first day contains methicillin-resistant Staph aureus. Okay. What I want to show to you is that the length, the duration that you are taking to identify the bacteria. So 6 days if everything goes on well, yeah. But you know, once you are in the lab, sometimes, you know, you don't have this ready, you don't have this ready, you don't have mannitol salt agar ready. So there might be hiccups here and there. So you might be ending up doing the diagnosis in 10 days time. Yeah. So in the meantime, you can think of what happens to your patient. Okay. So other systems, okay, there is now Vitex. Yeah, which is very useful if you have a large number of samples. You know, if your lab is receiving maybe, you know, at the most like 20 samples per day, so I don't think it is worth, you know, investing in this. So there are many other automated ID systems which is available. Yeah. And then, of course, all this analysis, you have to perform it in pure culture. Pure culture means, you still have to culture, and then it has to be contained, in a plate or in a solution. It contains only one type of bacteria. Yeah, so it has to be pure. Once if it is mixed, it is gone case. You won't be able to do your identification. All your results will be haywire. You won't be able to identify. Okay. So all this while, okay, I've been doing work in the lab since 1994. Yeah. So lab diagnostics for veterinary pathogens, we have relied mainly on culture. Yeah. However, for the last 15 years or so, there have been coming up with new molecular techniques. So the slides after this, I will introduce to you, what are the techniques which you'll be able to, apply and adopt, in your lab, to do identification. Yeah. And of course, here you can see here, it has broadened the scope of diagnostics, provide powerful tools that enable rapid and specific diagnosis in animal diseases. So you remember just now the slide number 3, just now, the flow, what are the ideal diagnostic tools that you should have? It has to be cheap, it has to be rapid, it has to be widely available. So that one you have to bear in mind. Okay, so usage of nucleic acid, based diagnostic has increased exponentially because over the years, the consumables is getting more affordable. Unlike in the 1980s, perhaps, for you to do polymerase chain reaction, per sample, it might cost you maybe 200 ringgit. 200 ringgit is about 70 US dollars. Yeah. But now, if you want to do PCR-based techniques, per sample, you might be able to carry out the test with maybe $2 or $3, per sample. Yeah. And the third point here, molecular diagnosis is most appropriate for pathogens that are, one is difficult to detect and identify in timely manner with conventional methods. So there's no harm for us to use molecular techniques for common bacteria, E. coli, Salmonella, Pseudomonas, Staph aureus. However, it is very, very useful if you are dealing with very, very fastidious organism. Fastidious means, it is very difficult for you to grow, very difficult for you to handle, and dangerous. Yeah. Like Microbacterium, Legionella. So it's very, very dangerous for you to handle. Yeah. So perhaps you can also have a look, some of the pathogens which is you are supposed to handle in BSL3, biosafety level 3. Yeah. So those are the things that you should consider, to apply molecular diagnosis rather than doing culture. Yeah. And then, of course, non-culturable pathogens. Yeah, so there are also some pathogens which is non-culturable, or anaerobic bacteria. Once you have a sample which you are supposed to culture in anaerobic, oxygen is very toxic for them. So you should use molecular diagnosis. Yeah. Okay, where can you find DNA of a bacteria? Okay. So I'm sure, you have gone through this basic cartoon of bacteria. So this is a refresher for you. So bacteria, they do not have nucleus, right? So it is a prokaryote. So differentiate between eukaryote cells and also prokaryote cells. So that one you have to have in mind, because, once if you deal with bacteria, it is a very simple cell. So it's, you have to understand the characteristics of a bacteria in order for you to troubleshoot or whatever, in molecular diagnosis. Okay. So if you see here, this is a very, this is a rod bacteria cell. Okay, so you can see here the pili is being used to attach to the host. You know, if it is causing disease, it has to attach first to the host before it multiplies and all. Yeah, and then you can see here that the genome of the bacteria is scattered in the cytoplasm. Yeah, there's no nucleus. You have to remember that. Yeah. So it's all in this cell. So if let's say you take out this DNA, if you take out this DNA, you can see it's all condensed, right? So once if you take out this DNA and then you stretch it out, it will be like over 200 times the length of the cell. So you can imagine like how cramped is the DNA inside the cell. Yeah, and you also have to see the other non-chromosomal DNA, which is called the plasmids. So these are the small ones. So you can see two here. So non-chromosomal DNA is not compulsory for bacteria. Okay, but many of the molecular diagnosis relies on plasmid, especially if you're working with antimicrobial resistance. Okay. Okay, so this is another cartoon. So this is much simpler. Okay, so this is a bacterial chromosome. So it's like, you know, spread around the cytoplasm of the bacteria. And then you can see here plasmids. Yeah, so plasmid, it can come in various sizes. But of course, you know, it is much, much smaller than the genome. Okay. Okay, molecular methods. So I've listed here some of the common molecular methods used. This is not exhaustive, so there are many more techniques which is available. And what I'm going to cover today is going to be polymerase chain reaction. Yeah, and then the next one would be the pulse field gel electrophoresis. And the last one would be multi-locus sequencing typing. So reason of me choosing or selecting these methods is that polymerase chain reaction is widely used. I think almost maybe 80% of the diagnostic lab, they will have this thermal cycler, they call it, you know, the machine where they use to amplify DNA. Yeah, and then pulse field gel electrophoresis is very useful if you want to do molecular epidemiology of the bacteria. Also very useful, and for some of the bacteria it is considered as gold standard. Okay, and the last one is the multi-locus sequencing typing. So this is the more recent method, and it is more widely accepted. So compared pulse field and multi-locus. So multi-locus is now more widely accepted. Yeah. However, it is not yet regarded as gold standard to differentiate bacteria. Okay. So afterwards I'll explain more. Okay. So what are the advantages of using molecular techniques? One, it's rapid. Okay. So these are the take-home messages that I like the students to remember. Okay, you compare conventional methods to the molecular methods. One, it is rapid, it is fast compared to conventional method. Yeah, it is more definitive. Okay, so if you have experience doing identification using biochemical test, many times, you know, when you do biochemical tests, you are not able to identify up to the species level. Okay, contohnya, okay, if let's say you are looking for salmonella, okay, so you will end up only up to the salmonella species, but you cannot, you know, go deeper. Is it salmonella enteritidis? Is it salmonella typhimurium? Is it salmonella pulorum? Things like that. Why? Because there are some variation between the strains which will not allow you to differentiate using, you know, your naked eye. and the biochemical test. Okay, so it is very definitive if you use molecular methods. More discriminating, so this one is quite related to the second point. So, it will be much difference, whether you have, mycoplasma synoviae or mycoplasma gallisepticum, something like that. Yeah, and a molecular methods is adaptable to all pathogens. So just now I was saying that, it's good, if you use these molecular methods to apply to all these fastidious organism. But with molecular methods, you are able to do it for all, whether it is a simple organism, everyday organism. E. coli is the most famous bacteria. So you can use it for E. coli. You can use it for microbacterium. You can use it for Brucella or whatnot. Okay. And for E. coli, perhaps, rather than just detecting E. coli, yeah, perhaps you want to identify pathogenic E. coli or, E. coli 0157. So all those things are possible. Yeah. So let us go to the first one. So this is polymerase chain reaction. I'm not sure whether you have gone through this technique or not, in your practical. So what does polymerase chain reaction does is that it is something like a photocopy machine. Yeah, machine post photocopy. So it makes millions and millions of copies of your targeted DNA. So just now you remember in a bacteria, just now there is the DNA, right? So it can make millions and millions of copies until you be able to see with your eyes, with your naked eye. Yeah. So if let's say it is so powerful until you can make billions of copies of the DNA, you can imagine like you can eliminate all these problems of not having medias and whatnot. Okay. So PCR, it is making copy and it is mimicking, it is following what DNA replication is doing in a host in vivo. Okay, and PCR allows millions or even billions of exact replicas of specific pieces of DNA to be synthesized from a single copy rapidly. So you imagine, let's say, okay, you have a fecal sample, right? So in feces, you can find a lot of different bacteria, right? So if let's say I want just for you to identify E. coli in the sample, okay? So even there is only one E. coli cell in the sample, PCR is so powerful that you'll be able to pick out that particular DNA and tell you, okay, there is there are E. coli in the sample. So it's so powerful. So even though there are maybe over 20 types of different organisms inside there, so if you're targeting for one bacteria, you'll be able to do so. Okay, so this is a diagram just to give you an idea. So this is the template DNA. Template DNA means this is the mother copy of the DNA. So in a bacteria, just now you see, the clump of the DNA, right? So you're just targeting a small part. You're not amplifying the whole chromosome, just a small part of it. Yeah, and then, you carry out PCR, and then PCR is replication of DNA. So it replicates 1 becoming 2, 2 becoming 4, 4 becoming 8. And after cycles, normally we will do like over 30 cycles. You will end up, you can see here, 35 cycles, you can end up having 68 billion copies. So from 1, you can get this many. And how long does it take from here to here? Is only a few hours. So it's very, very powerful. Yeah. Okay. So what do you need, if you want to do PCR? Okay. One is you have to have the source of the DNA. okay, is either a bacteria, a pure culture, or even a sample, directly from the sample. If you have a swab sample, if you have a feces, if you have organs and whatnot, so you have to have the template where the DNA of the bacteria is there. Okay, and the other things here is all the material where you need, you know, for you to carry out PCR. Yeah, so you put everything, of course, you know, with the proper concentrations, and then you just use, you know, it is for microfuge tube. So this one is even smaller, okay, for you to fit in the machine that you have. Yeah, so it's not the common 1.5 mils microfuge. So it comes in perhaps 0.5 mils or 0.2. So it's very, very tiny. So when you do PCR, we don't advise you to do only for one sample. So if you have 10 samples, you prepare a cocktail with all these things, this raw material, and then the end only you put the different templates, you know, in different tubes. And then you can see here, perhaps the picture is a bit small, but you can see here, this is strip. Yeah, strip. So each here, you know, it contains different sample. Yeah, and then once you close and on the machine, so the PCR process will carry out. Okay. Okay, then after 2.5 hours, 3 hours, when the process is done, after 36 cycles of the PCR, so how do you see whether your PCR is working or otherwise? So you will run it on an agarose gel. Okay, so this is the agarose gel where you load the different samples into different wells and you apply electric current, and then you will analyze the results. Okay, so this is the first lane here is the marker. So the marker normally is a ladder. Okay, because PCR, it is double-stranded and then it's linear. Yeah, so you can use ladder marker. And then here is the product. You remember just now, from one copy, it can make millions. So this is where you can see, you know, with your naked eye. So if, you know, you have a target amplicon, you know, on your gel, meaning, you know, that there is a bacteria that you're looking for in the sample. Okay, for types of PCR, many types. Okay, what I'm going to share with you, which I think will be very useful, which is the monoplex and the multiplex PCR. Okay, contoh So example, monoplex PCR. Oops. Detection of Salmonella species in a sample. Okay, so this is feces. Okay, so this will act as your DNA template. Okay, so you can take directly from the sample, or many times also people like to culture first and have bacteria growing on the plate and use that as a sample. Yeah, so once you have that, yeah, and then with all the other ingredients of the PCR, you load it on the machine, and the primers that you use, which will specifically identify Salmonella from all other bacteria, I'm giving you example here, is Sal F. F is for forward and Sal R is for reversed. Oops. Okay. And we are expecting a product size 500 base pair. Okay. So if you use this primer will go into the template and look for whether are there any Salmonella or not. Yeah, so it's very specific. So this is the example of gel that you will going to get. So this is the ladder. Yeah. So ladder, you can see here, this is 100 base pair, 200 base pair, 500 base pair, and 1000 base pair. Yeah. So this is the positive control. So positive control is you use a pure Salmonella culture and use it as template. Yeah. So you'll get a band here. And a negative control, perhaps you want to use other bacteria. Perhaps you want to use E. coli as a template, or you can also use perhaps distilled water as the negative control. Okay. So number 1, 2, and 3 is your sample. Okay. So you can see here from your results, sample number 1 and sample number 3 contains some Salmonella. Okay. While sample number 2, you can't see anything, no amplicon here. So that means the sample does not contain Salmonella. Okay. So that is monoplex. Monoplex means that you use only one set of primers. This primer is so powerful that you'll be able to detect, select and identify only that particular bacteria. Okay. Next example. So just now was monoplex, one primer, right? So the next one is multiplex PCR. Okay. So I'm using example for Salmonella as well. However, this time, I'm not going to look only for Salmonella species. I want to go deeper. Okay. So I want to identify whether are there any Salmonella enteritidis and Salmonella typhimurium in the sample. Okay. So we still have the sample. You can culture. Yeah. Perhaps some of you who might have experience, you know, if you have a sample which is so I can say it's a complicated sample because most of the time they will have other debris, right? So you culture on a media and then you take that as a template. Or, you know, if your sample is a swab sample, you know, is quite a clean sample, so you can use that directly. Okay, and then you run the PCR. Okay. The difference between the monoplex and the multiplex is that you use more than one set of primers. Okay. So you use, sorry, yeah. So this one is supposed to be, okay. Salmonella, okay. There is some typo here. So perhaps afterwards when I give you the slides, so this one you have to change into Salmonella E. No, ST, okay, ST here rather than SF, okay. So you have here Salmonella, wait, let me see here. Okay, this is ST, this is SE. Okay, ST stands for Salmonella typhimurium, SE stands for Salmonella enteritidis. And if there is Salmonella typhimurium, you will have 500 base pair. If it is enteritidis, you will have 250 base pair. Okay. Instead of one set of primer, you use two sets of primers. But the thing is the same. Same sample, the same cocktail, only that you add in another primer. So you use two primers. Okay. So you put it in your machine. And this is the results that you get. Okay. So in sample number 1, okay, the positive control here, 500 is for ST, right? Just now, okay. So in the positive sample, you can get ST as well as SE. So there are both bacteria there in your positive control. In your negative, you can't see anything. Yeah. So for sample number 1, there is ST, there is SE. Sample number 3, only ST, right? Because this one, there's none here. So sample number 2, there's no Salmonella enteritidis and there's no Salmonella typhimurium. Okay. So I hope that that is clear. Okay. So if you are not clear afterwards, perhaps we can come back and discuss this one. Yeah. So this is the diagram. Yeah. So you see here, this is a multiplex PCR with all 4 primer set. You can see all the different colors here, right? A, B, C, D. Yeah, with one template, and then you can detect whether if they are gene A, so you show the red amplicon, yellow amplicon, green amplicon, and the blue amplicon. Okay. Okay, I'm going to share with you some of the work that we have done here in UPM. So the first one is we did some work on Campylobacter. So a Campylobacter is a very fastidious organism, right? Yeah, so it requires carbon dioxide, and then it's very difficult to grow. So this has been done quite some time ago, and this study we used multiplex PCR. Okay, you can see here this is multiplex PCR, and you can see here this is the different primers that we have used. Set 1, set 2, 3, 4, 5, 6, 7. Okay, so you can see how powerful multiplex PCR is. It does not only detect whether are there any Campi or not, it can straight away tell you what species they are. Okay, so let us see the results. Okay, so this one I think is a better example than the one that I showed you just now. Okay, let us focus on this band. Yeah, you can see my arrow, right? So this one, 816 base pair. Okay, lane number 1 and lane number 18, this is the ladder. Yeah, so 816, this is the genus Campylobacter. So we can see, okay, sample number 2, 3, 4, the ones that, it highlights giving you the amplicon, there is Campi there. Okay, and we go a bit below, 735, it is a Campi jejuni. So for lane 16, okay, it is a Campylobacter, and what Campi is it? It is a Campi jejuni. Okay, let us go to lane 13. So yes, it is a Campi, and it is a Campi Larry, 251 base pair. Okay, so it is similar to the others. So it's very powerful. Okay, one question. Can we have a band with the species but the genus does not highlight? If let's say here, if this one is not a negative control, if we have one band here, is it possible? It won't be possible, because in order for it to become a species, it has to have the genus first. Yeah, so it has to be Campi. So if let's say your results, if let's say you're doing research and then you are inventing all these different primers, yeah, and then when you run your sample, suddenly for this Campi, suddenly you have here 251 nicely having here, but your genus is missing. So something is not right with your PCR. So everything becomes invalid. Okay, so I hope you understand that, with multiplex, it's very powerful, but you have to be careful. Once if you have something with the genus, all the species, it has to have the genus first before you have the species. Okay, so this is a good example. So if let's say you want to revisit, so perhaps this will be a good one. Yeah. So the next one is the MRSA. So phenotypic and genotypic characterization of MRSA isolated from dogs and cats in University Veterinary Hospital in UPM. Okay, so this study was done in 2010, a long time ago. Okay, so this one is simpler. So this is a monoplex. Yeah, just now was multiplex, this is monoplex. So we just wanted to identify, are there any MRSA or not in the sample? So we have the sample, we run it with a MAC-A gene. So you can see here in the right up here. So it's a MAC-A gene because all MRSA, they will have this MAC-A gene in the genome. Okay, so if you don't have any of this, so you can say that is not a MRSA. Okay. Okay, let us again revisit this MRSA. You have samples from poultry. Conventionally, you will culture on blood agar. You select on mannitol salt agar. You reselect on OSAP media. OSAP media is a selective media for methicillin resistance. Yeah. And then you do antibiotic susceptibility test in order for you to find that multi-drug resistance characteristics. Okay. And then you interpret the AST. So everything goes well, it requires you 6 days. Okay. And if you opt to do molecular PCR, what you need to do is, you culture, and then the next day you do PCR. PCR, you can just do it in a day. Half a day would be enough. And you can get that results 2 days. Or, if you have a method how you want to clean your samples correctly and whatnot, you can do it within a day. So you can see, how powerful PCR is. And this can be applied in your lab, diagnostic lab. Okay, so that is the first part of the lecture. Dr. Aan, do we invite questions now or will we wait until the end?
It depends on you, Prof. If you want to going to the Q&A session now, is it okay? So
I'm halfway through. I'm halfway through.
Yeah, I think we can finish all the presentation first, Prof, and then we have a Q&A session after that. Is it okay, Prof?
Okay, sure. So this one is
Prof.
sharing you just a photo. Yeah, so this one is our faculty, and this is the academic building. Yeah, so just to show you, how our faculty looks like. Okay, then let us move on. So I finished with the PCR. So let us see, a scenario. Nowadays outbreaks is happening, every now and then. So how do we detect the organism? Where does the organism originate? So those are the questions which comes into our mind, right? And what is the spread of the organism? Yeah. So how do you answer that? Yeah. Can we do PCR or how? Yeah. So the answer is, if you want to do molecular epidemiology, yeah, one of the powerful technique that you can use is pulse field gel electrophoresis. Okay. So just now for PCR, the gel that I showed you, that is a conventional gel electrophoresis. Okay. So there is a limitation whenever you run a conventional electrophoresis. It cannot dissolve large fragments of DNA. If you go beyond maybe like 400 bases or whatnot, you can't really differentiate the DNA. It becomes stuck in the gel. Yeah, so it cannot be separated. Once if you cannot separate, you can't make sense out of the results that you can see on the gel. What you can see is just a large smear. Yeah. So in 1984, so most of you were not born yet that time, I guess. Yeah. So these two scientists, they invent what they call the pulse field gel electrophoresis. Okay. So let us see what does this thing then do. Yeah. So pulse field, unlike the normal gel electrophoresis. Okay. So gel electrophoresis, you have a gel, and then the current goes only in one way. Okay. But for pulse field gel electrophoresis, it goes in 2 orders magnitude. So you go right, you go left, you go right, you go left. By doing that, this scientist found out that it can separate large fragments of DNA better. Okay, so this is the key of pulse field gel electrophoresis. Yeah, so you can see here this note, pulse field resolve large DNA. Yeah, so raising the upper limit of the DNA separation to over 10 megabase. Okay, so of course, if you want to study epidemiology, you wouldn't want to be based only on small fragments. Just now, for detection is fine. Are there any E. coli? Are there any salmonella? You detect, right? But if you want to study the molecular epidemiology, you need the whole genome. Yeah, so if the whole genome, if you run it on a conventional gel, you won't be able to resolve the fragments. Okay, so pulse field can do that. So that is the advantage of doing pulse field. Okay, so if you search about pulse field, you can see nowadays people are more inclined to use pulse field if they want to do molecular epidemiology. So you can see here the example, CDC, they use pulse field to study disease outbreaks. Yeah. And the second point and third point is to show you why, you cannot run the DNA, the genome of the bacteria in a conventional gel. Okay, because large DNA, it is easily sheared. Sheared means broken, pecah. Yeah, because once if you extract out, from a bacteria, you extract out the DNA. Once if you tilt the tube or whatnot, the DNA will break. It's very, very delicate. Okay, so in pulse field, we don't extract out the DNA and put it in solution. What we do is that we put organism in a gel. So you see here in pulse field, organism is embed. Embed meaning, you have a agar and then you put the bacteria in the agar. It's not on the agar. So the agar is still molten. It's still chai, it's still very molten. Of course, you cannot use, high temperature. So it is about 45 degrees. And you put the bacteria in the agar and then you treat the plugs. You treat the bacteria in the gel itself. By doing that, the bacteria is safe, meaning the conformation of the DNA is undamaged. Okay, so that is the difference between the gel electrophoresis and the pulse field gel electrophoresis. Okay, so this is the flow. Yeah, so you have the culture on a plate. Yeah, and then you multiply the bacteria. Yeah, and then what you do is that you have to adjust to a certain concentration, the McFarland standard. Yeah, so McFarland, I'm sure you have used it, 0.5 McFarland. So this one is a higher concentration of bacteria in the solution. Yeah, and then this one you put it in this little wells with agar. Okay, so this is what we call the plug preparation. Okay, this is another explanation. So you imagine just now you have that bacterial suspension, McFarland 6, right? Okay, and then you put the bacteria together with the agarose, yeah, the gel, the agar. And then the bacteria now is in this small little agar cubes, plugs, we call it. Yeah. So in this small, this one, and then you run it on the pulse field gel electrophoresis after you treat it with enzymes. So the job of these enzymes is that they cut at certain places of the genome. Okay, and then the pattern which is being produced by this restriction enzyme will indicate what strain of the bacteria they are. Okay. So this is the one which I mentioned just now, the alternate current that is being used for pulse field. You see here 120 degrees. So the shape of the gel is still the same. It's still rectangle, but then the machine itself, the pulse field, it is such a way. Yeah. So you can program, you know, at you say like every 30 seconds, move the current on the right. Every 1 minute, you move the current to the left, something like that. And it has to be carried out in cold temperature. So you see here at 14 degrees. Yeah. And the electrophoresis takes a long time. So you imagine, because the genome is very big, so you need a long time, and normally it runs overnight, 18 to 24 hours. If like just now for detection using PCR, you just need maybe 2 hours, right? So this one needs much, much longer because you are dealing with the whole genome of the bacteria. And what you can see, the results is that these are the patterns being produced by the different strains. And these different strains will show you, okay, like S1. S1 is similar to the strain in Thailand, for instance. So you can say, oh, perhaps this outbreak has been caused by, you know, the strain which has spread from another country to our country, something like this. Yeah. Okay, so this is another example for the current direction of DNA migration. So you can see here, left, right, left, right, left, right, compared to the DNA migration in the conventional gel electrophoresis. Okay, and as I said just now, many organisms have not many. Okay, people, CDC or OIE, in fact, you know, they have regarded this pulse field as the gold standard to differentiate the strains of the different organisms, outbreak studies, yeah, using for outbreak studies. Okay, so this is one example of the gel which is being produced. So you have to also imagine just now the different usage of that PCR detection and with this pulse field. This pulse field, you will be able to see more bands. Yeah, more bands because you want to differentiate the strains according to the location. Okay. Okay, so this is another schematic diagram. Okay, so there is a software where you can use and you can align, you know, the different gels, you know, because one gel, it can take up only to 15 samples. So if you have like over 100 samples, so you need to run multiple gels, and there is a software which can do that. And it can also analyze for you, and it will clump. Yeah. So you can see here the different numbers and all this. So you say, oh, you know, our strain here in Surabaya is similar to Jakarta, but different from Bali, something like that. Okay, so it's very powerful in that sense. Yeah. So these are some of our work which have done with pulse field gel electrophoresis. So we did some work on Arcobacter. Yeah. And then we also did some work on MRSA. Yeah, with a pulse field. And then, oh, this one is one example, characterization of Campylobacter. So this is not our work. Okay, so this one is one. This is our work. Molecular epidemiology for this is for MRSA. Yeah. Okay, so that is the second part. So let us move on for the third part. So we have covered just now PCR. And then we have covered the pulse field gel electrophoresis. So the last one is multi-locus sequencing typing. So similar to pulse field, so if you Google and find papers for pulse field gel electrophoresis, perhaps 5 years back is very, very widely used. Okay, but in the recent years, we have seen researchers now evolving and using MLST compared to the pulse field. Reason one is because pulse field, you need a specialized machine. Okay, a specialized machine, it will cost you, let me calculate, it's about 50,000 USD. Yeah, and then that it excludes the software. So the software will be about like 20,000 USD. Yeah. With the MLST, a multi-local sequencing typing, you just need a PCR machine. So the one that you use for your diagnostic, for detecting of your for pathogens just now, you can apply that and use it as MLST. Okay, so point number 2 here, you can see it is portable, reproducible, and scalable typing system that reflects population and evolutionary biology of pathogens. Okay, so it can serve about the similar purpose as pulse field. Okay, and the other advantage of MLST is that if I do it here in Serdang, and you know somebody in UNER does the MLST, the results will be the same. Okay, compared to pulse field just now, I do with my machine and Dr. Yola do with her machine there in Surabaya. Sometimes, you know, there will be discrepancies because the even though we use the same brand of machine, the consumables might be different. The solutions prepared might be different. Perhaps she used 13 degrees. I use 13.5 degrees. So there might be some minor variation. So people are now, you know, more inclined to use MLST compared to the pulse field. Yeah, it's very robust, as I said just now. Even though, you know, somebody in India, somebody in Europe does this, it will be the same. Very robust, efficient method in exploring the molecular evolution as well. And it is easily shared between labs because the database and how you analyze it is free online. Okay, so it is a very powerful thing. So this one is the example that I'm showing, you know, why, you know, people are moving from pulse field to MLST because of the inconsistency. Yeah? Different solutions, different temperatures, different brands of machine. Sometimes it is a bit inconsistent. Yeah, so people are moving towards the MLST. So how do we do MLST? Okay, so MLST is based only on housekeeping genes. So housekeeping genes of bacteria, it is all located in the genome. And for every bacteria, every type of bacteria, they have selected 7 housekeeping genes. Yeah, and you can Google and see this database. Yeah, so you know, we have conducted a few courses, you know, how to use MLST, and I guess like most of our participants are very happy, you know, with what they can get with MLST. Yeah, so what you need is identify the housekeeping genes. You use PCR, and then you analyze using the free database, and they will give you, you know, an allele. And this allele, when you add up, it will give you a sequence type. Okay, and with the sequence type, you'll be able to do your molecular evolution of your bacteria. Okay, so some of you might be asking, what are housekeeping genes? Yeah, we know housekeeper, right? Okay, so this is housekeeping genes. So even bacteria, they have housekeeping. So they do this housekeeping. So they are the genes that are required for maintenance of basic cellular function. So they are more or less conserved in organisms. So that is why it is being used, you know, to analyze organisms. Okay. How do we do that? Yeah, so it's very simple. So even I also, I'm very happy, you know, if we are asked to do MLST compared to pulse field. It's less laborious. Okay, so you have fresh culture, you do DNA extraction. Okay, and then if you want to confirm, normally we do confirmation, yeah, with PCR, and then you do MLST with amplification of 7 housekeeping genes. So one bacteria, we need to use 7 housekeeping genes. And where do you get this 7 housekeeping genes? Is all in the database. Okay, so that they will tell you, if you're working with Staph aureus, you use this gene, ARO-A, TPIL, you know, they list down. If you want to use MLST for leptospira, so there are another different sets of housekeeping genes. Okay, so after that, after doing the PCR, it's just a normal PCR that I showed you earlier. And then you run it on the electrophoresis, the normal gel electrophoresis. And then those amplicons that you get, you will send it for sequencing. Because the size of the amplicons are not big, it's about, you know, at the most it's 600 base pair for each of the housekeeping genes. So you can do it in only one run of sequence. And then from there you analyze, yeah? And what you get, you remember just the 7 housekeeping genes? So this is Staph aureus, okay? So this is the housekeeping genes, the names of the housekeeping genes. ARC, ARO, GLP, GMK, PTA, TPI, and YQI. Okay, and the primers you can get it from the database. Okay, after doing PCR, okay, you send it to the database. The database will give you a number, so you give you 1. Okay, and then with the same sample with the second housekeeping gene is also 1, and then different housekeeping genes. So all these numbers, you will input it in the database. They will give you the number. But this one is coincidentally, you know, it is the same, yeah. But you know, it can go different numbers as well. So in the end, the sequence type is 1. So 1 plus, it's not plus, yeah. So this one combination with 1, 1, 1, it will become 1, yeah. So you can see here there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 Staph aureus. All samples come from wound, yeah. But 8 samples are of the same sequence type. So this one you can see here, sequence type 1, the geography location is Australia, Canada, Gabon, Bulgaria, Japan. So you can see the widespread of this strain. Yeah. However, you can see here, this is the unique one. Yeah, the sequence 11 is also from wound. Yeah. ARC, the LL is 6. ARO, the LL is 79. So on and so forth. And the sequence type is 177. Yeah. So you see here, sequence type 177. It has been only reported in UK and US. And it was first reported in Asia and Malaysia. So the rest is all common, is widespread. But this one is very unique. So where does this one comes from? And then we can see, okay, sequence type 177 have been reported in UK and US. So everything is there in the database, yeah? And from your data just now, that is in the form of table, right? So most of the people, they don't like tables, you know, because it's difficult to see, difficult to analyze. So they like something which is more graphic. So this is called what is called is eBurst, EBURST. Yeah. So even this is free. You know, you can do it in the database, in the software, and it will give you this nice. What do you say? It's like a bunga api. Yeah, it's like a fireworks. Yeah, and you can see here, 78, for instance, here, you can see that the circle is big, right? So meaning there are so many reports having sequence type 78 compared to ST16, the size is smaller. And you can see here all these little black dots. So it represents the different numbers of the isolates, the strains which is available. So the further it is, the less related it is to the bigger clonal complex here. So you can see, like at one go, you can see here, this is very far, right? So this is a simpler one. Yeah. So ST21. So let's say, this ST21 is the main ST, sequence type available, in the country. So you might see this is the most, the biggest circle that you find. And then perhaps, this small one is the one that you recently find. So, okay, now you know that this one is very unique to you, and it does not, correlate that much with the main codal complex. Okay. So there are many things that you can interpret using this e-bursting. Okay. So we did some work as well with MLST. Yeah. So MRSA. So mainly you can see my work, I focus more onto MRSA. So this is my last slide. Yeah. So just to recap again, the learning outcome of this lecture is that I want to introduce to you some of the techniques, the useful techniques that can be applied, for your diagnostic as well as for your search. One is the PCR. I think is very useful for diagnosis, and then for research, for pulse field gel electrophoresis as well as the MLST. Okay. So I guess terima kasih. That's it. Dr. Aan.
Yes, Prof. Yeah,
Did I
actually,
go too fast?
No, Prof. But actually, you still have 20 minutes left. But it's okay.
Okay.
So we are now going to the
I can
Q&A
invite
session.
questions.
Yes, Prof. No.
Or is it after Prof. Aini's?
Yeah, I think it's better for having a Q&A session now, Prof. Is it okay?
Boleh.
Boleh. Okay, so we are now going to the Q&A session. So for the students who want to ask the question, you can ask directly to Professor Zunita related to the topic. Yeah, so you can raise hand and then you can ask directly to Professor Zunita. This is your great opportunity. You can ask directly to the expert. So please, for the students. Hello? Who's speaking now? I think Dr. Saif.
I can't really hear.
Yeah, I think Dr. Saif, I think your internet is unstable now.
Perhaps they can write the question in the chat box. That is also possible, right?
Oh yeah. Yeah. This is also, that's possible, Prof. If you have any question, you can directly ask to Professor Sunita, or you can type your question in the chat box. Yeah, Dr. Saif, I think your internet is unstable now, so you can type your question in the chat box. Okay, while waiting for Dr. Saif typing his questions, maybe for students who want to ask the question, please, you can raise hand and introduce yourself.
Malu mungkin.
Okay. Yeah. We have one question from Bernadette Caroline. Yeah, Bernadette Caroline. Yes, you can start to ask the question.
Good afternoon, Professor Zunita. I would like to ask some questions. Yes, my name is Bernadette. So my questions are, in PCR, do you make the primer by yourself or is it made by a company, and what is the primer made of? And that is my question.
I can't really hear you. If Dr. Ann can you repeat the question for me?
Bernadetta, ya? Bernadetta, can you repeat the question? Actually, we cannot hear you clearly.
Can I type the question in?
Yes, yes. Please just type, I guess. Yeah.
Yeah. Okay. Okay, so I will read for you.
Okay, so
Professor.
she's asking, yeah.
Okay.
Do we make our own primer, or is it made by a company? Yeah. So if you have a synthesizer in your faculty or in your university, you can do it yourself. However, it is much faster, you know, if you just ask you know, a company to do the primer. So what you need to supply to the company is only the sequence of the primer. So you might also ask, where do you get the sequence of the primer? Because just now I gave you some examples. Salmonella use this primer, E. coli use this primer. So you need to have, you know, some literatures, and, you know, if you want to say detect E. coli from swab sample. Okay, so you can Google and see E. coli primers. From there, you can pick up the sequence of the primer. So normally it's very short. It's about 20 base pair. So you just send that sequence to a company, you know, molecular biology company, probably, yeah, and then they will synthesize for you. And when it comes to you, it will be in a freeze-dried form. You can't see anything. Okay, so once you receive that primer, normally it'll be in a 1.5 ml tubes, right? So before opening it, yeah, you have to spin it down in order, you know, for the freeze-dried things to settle down in the tube. You know, I had my own bad experience when I first did my master's. I didn't know that, you know, the primers is freeze-dried. Yeah. So when the company sent to me the primers, I can see empty tubes. I can't see anything. So I just open up. So all the, you know, the freeze-dried materials, it goes off. So spin it down. Yeah, and then do your calculation in order for you to know like how much of distilled water for you to add in the primer. Yeah. So that is for your question. Okay. And then Dr. Saiful Rahman, how about sequencing?
We have another
if
question.
we work on virus or bacteria like that? How do you compare with other countries? Which countries is best for sequencing? If we work on virus or bacteria in Indonesia, how? I don't quite understand the question, Dr. Saiful. Saif.
Yeah. Dr. Saif is a PhD student from Pakistan.
Okay.
Yeah, but I think his internet is unstable. That's why he cannot ask directly.
Okay, perhaps I can just explain to him. Okay, he's asking about how about sequencing. Okay, sequencing, meaning you have to do your PCR in your own lab, and then you need to send that material for sequencing, to a company, unless if you have a sequencing machine in your university. Okay, you have to send it outside. Yeah. And then they will give you, after a week or two, they will give you sequences in chromatogram and then also as a words format. And then from there, you can do your analysis. Okay, and then he asked here, how do we compare with other countries? Okay, Dr. Saif, there is many software, how you can compare, your genome with other countries. There is a lot of software available. Which countries is best for sequencing? Anywhere. Now, perhaps what you can do is that you can outsource it to a company. Of course, now you are in Indonesia. If you engage with a company in Indonesia, they will advise you accordingly. Like for us in Malaysia, sometimes we also send it to Korea. It's not because of the quality, but most of the time is because of the cost. Yeah. I think quality wise, I think many of the companies they are at par now. Yeah? They give good results.
No. In previous publication, I have read some papers. And
Sorry, Dr. Saif. Dr. Saif.
Okay,
we cannot hear your
write.
voice. I'm sorry. Yes, please write it.
So he has another question. Is it?
Yeah, is there any more question from other students?
Okay.
Untuk mahasiswa semester 3. Ayo, silakan yang ingin bertanya.
Kelas internasional, silakan bertanya.
Ya. Boleh bertanya pakai bahasa Melayu ya, Prof? Prof Zunita?
Boleh.
Boleh.
Boleh sangat.
Boleh sangat.
Saya juga boleh practice Indonesian.
Mungkin mereka malu-malu, Prof.
Jangan malu-malu.
Malu-malu bertanya. Ayo adik-adik yang mau bertanya bisa pakai bahasa Indonesia aja, nggak apa-apa. Silakan, ini kesempatan yang baik untuk kalian karena bisa bertanya langsung dengan expertnya.
Ya, perwakilan dari kelas A, kelas B, kelas C, kelas D, kelas E.
Banyuwangi, mahasiswa dari Banyuwangi juga join kan ya? Okay, we have one chat.
Hello, good afternoon. I got some questions.
Okay, that's clear.
Hello, Sarfin. Yes,
So
Sarfin,
good
please ask your question.
Love your presentation there. There's some questions I want to ask. Okay, on my cam.
Yes, please proceed, Charvin.
Oh, okay. Um, okay, if you, sorry,
Yes, Arvin?
should I type out? The voice is
Uh,
breaking up like that.
It was clear just
Sorry,
now.
can you repeat your question? Yeah.
Okay,
Yeah, you were clear just now, but now we cannot.
sorry, background, sorry. What is it again? Can you repeat, Dr. Yolena?
Yeah, we cannot hear you clearly. Can you repeat your question,
Okay,
Sarfin?
I'm still a little bit confused when it comes to the PCR, yeah? Can you explain a bit more about this multiplex? Thank you.
Apa soalannya tadi?
Sorry, Sarfin. Sarfin?
Yeah?
Sarfin, yeah. I'm sorry, I cannot hear you clearly. Can you type your question?
Okay.
Can you type your question?
1 minute, yeah?
Okay.
Okay, sorry.
Less than 1 minute.
Okay, 1 minute, yeah?
1 minute is too long.
Okay. Sorry. Yeah.
You were clear just now, but suddenly your voice like missing. Okay, Sarfin, 30 seconds left. Okay. Sarfin?
Sorry,
Okay,
I was
from
typing a
Sarfin.
question. By the time you asked, it's a bit long.
Okay,
Here's the
what
question.
do you think
What do you-
about this multiplex PCR method?
Sorry, I was typing a question. By the time you
Okay,
asked,
can I just read the question? Somebody else is talking, but anyway, so he has asked about multiplex. Do you think it is accurate than the normal PCR method? Multiplex PCR and monoplex PCR, they are normal PCR. Okay, there's no abnormal PCR method. Okay,
Oh,
so perhaps yeah, so I don't quite understand when you say the normal PCR method. Both of the multiplex and monoplex, they are accurate. Both of them are the same. The difference is that for monoplex, it uses only one primer, one set of primer. And for multiplex, it uses more set of primers. It serves different purposes. Okay, so both of them are accurate. You know, there's no doubt in that. Yeah.
okay. Thank you.
Is that okay,
And
Sarvi? Okay.
That's good. Thank you. There's something else. Something you mentioned about Korea. Can you explain more about it, please? I didn't catch it. What you meant?
Oh, okay. For sequencing.
Something you mentioned.
Oh, just now I just
you send it to Korea. Something like.
Okay. So for sequencing, okay, normally we outsource. We don't do our own, right? So I'm suggesting that if you don't have a good company, sequencing company in Indonesia, which I doubt, I think all countries, they have good ones, we can outsource it to Korea. because Korea has they used to give very good service, and they, you know, the turnaround time is very fast. You send to them your PCR product, or you know, your DNA that you want to sequence. Within 2 weeks you can get results back to you. We also don't want to send your our DNA, you know, to a company which they will sit down on your DNA for a month and then only you get the results, right? So we last time we used to send it to Korea outside. You know, out of the country. Okay, good afternoon. I have a question about PCR. I read about viral identification that use PCR test, and there is viral purification. Is there PCR test for bacteria step 2? Okay, for bacteria, afterwards, Prof. Aini, you know, she will go through the viral molecular virology. Okay, for bacteria, you have an option. Okay, so it's either you do your PCR directly from the sample, whereby you only need to break up the cells in the sample. Okay, how do you do the breaking up of the cells? Have you heard about detergent, SDS or something like that? Yeah, so you just add in a bit, you Votex, and then all the cells, you know, it will break up and all the DNA will be out in the solution. That is one way. Okay, the other way is that you do purification. If you do purification, meaning you can save your DNA in fridge, you know, in your freezer, and then use it maybe next week to do PCR. But if you are doing PCR directly from the sample, it is very advisable for you to do directly. You know, you take the sample, you process it, and do PCR on the same day. You cannot save it. Let's say, you know, you have your sample, you want to do your PCR in 2 weeks time. Okay, so you have your sample, you put it in the fridge. That cannot be done. Okay, because once if you already treat your samples, yeah, there will be DNAs, DNASE, where this enzyme, it will eat up your DNA. So in the end, you will get a false results. Okay, so I repeat again, you have an option. If you want to do PCR directly from the sample, advisable, do it in the same day. Or if you want to do your PCR later, you know, you go and collect samples from farm A, 10 samples, farm B, 10 samples. You want to you want to do your PCR at one go. So isolate and purify and save it. So because once you purify, you do not have this enzyme that can eat up your DNA. Okay, so does that answer your question, Izanti?
Yes, Professor, thank you.
Dr. Yolanda, I got one more question.
Yes.
I'm also thinking.
Yes, please, Arvin.
Should I type out the question, or should I type out the question or speak it out?
No, you can speak it now.
Okay. When the Prof mentioned about this ideal diagnostic method is cost effective. How much? What do you mean by cost effective?
Cost effective meaning it has to be cheap. Cheap, but then it serves the purpose.
How much?
We don't want something, we don't want, you know, a diagnostic test which is cheap, but is giving you false results, right? So the ideal method that we want, you know, it has to have all those elements. Yeah, it has to be fast, it has to be cheap, it has to be widely available. You know, even though the method is cheap, but then it's very difficult for people to get that method, yeah, to get source of that method. So it's also not possible. Yeah.
Okay. As you mentioned about cost effective, usually when you do all these tests, how much will it come up to?
It varies. Yeah, but as I said, you know, during the olden days, if you want to carry out a PCR test, you know, some of the consumables of the PCR is very expensive during the olden days, and it can come up to, you know, per test maybe 50 ringgit. It's about like $15, something like that. But nowadays, most of the consumables is getting cheaper and cheaper. Yeah, so perhaps it's like $3, you know, per test. Yeah, so I think it's you know, your clients would want to is happy to pay for that fees, PCR.
Thank you very much, Prof.
Welcome.
Thank you very much,
Excuse me.
Prof.
Doc, I wanted to ask a question. Can I ask in
Okay,
Malay,
Tarsan.
if you don't mind?
Boleh.
Boleh.
Okay. Is it just me or there's like background voices? Okay. I wanted to ask, kan kalau PCR, kan sometimes result dia macam tak tepat tau, ada yang macam betul salah kan. So like I just wanted to know like what are the possible reasons boleh jadi like result yang palsu gitu.
Apa kadang-kadang tidak tepat, macam mana Tarshan? Your question tadi?
Because I heard kan sometimes ada PCR yang orang buat PCR test or something like that kan kadang-kadang like result dia kadang-kadang tak tepat ataupun keluar result yang salah gitu kan. So like nak tahu apa yang possibilities criteria yang menyebabkan sorry kalau soalan salah.
Okay, so Tarshan is okay, Tarshan is asking about what are the possibilities of us getting false results from PCR, right? So most probably it will be from poor primer design, most probably. You know, if you use a primer which is not really that specific, you will get all these false results coming out. So if you go back to the slides just now, you can see, you know, one of the example I used for the Campylobacter just now, right? So there is no way for you to get a band with a species, let's say for jejuni only, without the Campylobacter genus being there. So it means that, you know, it's poor design, primer design. Okay. But most of the time, you know, it's very accurate, and then you can always troubleshoot, you know, with PCR.
Limited time, can we continue with the next presentation from
Okay,
Professor
saya boleh mula ke? Ada soalan lagi untuk Prof. Zunita?
Professor Zunita.
Saya dah boleh mula ke, Prof.
Okay. Yeah.
Okay.
Tak bagus.
Prof. Aini. Hello.
Boleh.
Sorry,
Tak
boleh
dengar.
dengar?
Saya kurang dengar.
Yeah,
Bu Ana.
sorry, Prof. I think my internet is unstable now. Can you hear me now, Prof?
Yes, I can hear you now.
Yeah, I'm sorry, Prof. May I read your CV first before you start the presentation?
Okay.
Okay, thank you, Prof.
Boleh.
Okay, thank you, Prof. Okay,
Right.
I would like to read briefly about the CV of Professor Aini. So currently, Professor Aini is a professor of Department of Veterinary Clinical Studies, Faculty of Veterinary Medicine, University Putra Malaysia. She was the Deputy Vice Chancellor for Academic and International University Putra Malaysia. She is also Chairman and member of various committees at many degrees. Graduated with Doctor of Veterinary Medicine from University Putra Malaysia, and she pursued her postgraduate study at her Master in Veterinary Science degree in Avian Medicine. And she obtained her PhD degree, continued with her post also at Cornell University, USA. And Professor Aini's research interest is in avian respiratory and immunosuppressive diseases. And Professor Aini has received many national and international honorary and awards throughout her career as a lecturer. 3 minutes presentation time, Prof. Thank you.
Ya, dah sudah ke? Suara.
Halo? Prof? Iya, sudah, Prof. Sorry,
Boleh dengar ya? Suara saya boleh dengar?
Yes, we can hear you clearly.
Okay. So thank you very much, Dr. Aan. Very happy. I'm very happy to see all of you again today. Thank you very much to Dr. Jola, Professor Yola for the invitation. And of course to the Dean, the new Dean, Prof. Dr. Mimi, for the invitation to talk to the students and to share some of the information, some of the work that we do with this on virology. I also saw just now my good friend, Professor Rahayu. Assalamualaikum, Professor Rahayu. Very happy to see Professor Rahayu and all the rest. Okay, Prof. Zunita has spoken a lot about the molecular aspect and also the process itself of doing the technology of molecular work, especially on PCR. And a lot of question has been asked about PCR. One of the question that was asked just now is that whether it is the same with virus. It is the same. Okay, the technology or the methods that can be used in virology and also in bacteriology, they are the same. For virology, usually in PCR or in detection of sample, we do purification first. We want to do it quickly and large sample can be used. Purification is the way that we do it. And of course, it has to be done quickly as well, not to keep for a long time before you do. And the best is to take the sample and to start right away. So that answers the question that someone asked just now. I think Izanti asked whether it is the same or not. It is the same. It's, in general, the methods, the molecular methods that are being used for virology and for bacteriology, they are the same. Maybe in certain aspect there is some slight modification in certain areas, but otherwise the principles are the same. So today, in my talk today, I'm not going to talk about the methods of doing the various techniques in molecular biology, because my area is more of applied application. So I will talk about the applications of molecular biology in the diagnosis as well as in the preparation of vaccine. So that will be the way that I will talk. But I will go through quickly what are those molecular methods that we use in virology. And I'm sure all of you have gone through the lectures on virology, and you're very familiar with what I'm going to say. So it's going to be a revision for you. And if there's anything that we can discuss about this application, that's where that my target for the lecture today. Okay, so my lecture today is more of the application of the methods in molecular biology rather than explaining about the methods itself. Prof. Zunita is a bacteriologist and she has explained in detail about the methods. If you look at those methods, they are similar to one that will be applied to virology. So that's why I'm not going to talk about the methods itself, but it's just the name of the methods and how it can be applied and how it has been applied in diagnosis as well as for vaccine preparation. Okay, good afternoon everyone, and selamat tengah hari, selamat sore, salam sejahtera, assalamualaikum warahmatullahi taala wabarakatuh. I hope that with all the situation in the country now with COVID, we are, we are in good condition. Everyone is in good condition. A little bit about UPM, University Putra Malaysia, because I'm sure not many know about University Putra Malaysia. Not many have been to University Putra Malaysia. That's where Professor Zunita and myself come from. Okay, we started as a School of Agriculture, Malaya in 1931. Then in 1947, it was upgraded to College of Agriculture, Malaya. And in 1971, it was upgraded as a university, University Pertanian Malaysia. Pertanian is, as you know, is agriculture. Okay, UPM is agriculture based. Our niche area is still agriculture, but we had to change our name due to the situation in the early years that we were not offering only but also other programs. When Universities Pertanian Malaysia started in 1971, there were 3 foundation faculties: Faculty of Agriculture, Faculty of Veterinary Science, and Faculty of Veterinary Medicine and Animal Science, and a Faculty of Forestry. So those were 3 faculties. And as the university progressed, more programs come in, but most of them are still agriculture based. But then there is a need for the country to produce more doctors, more graduates in other programs. They are not just agriculture based, and we offered medical program as well. So there was a need to change the name, and we changed it to University Putra Malaysia in 1997 after the approval of the cabinet. Yeah, just bear with me a little bit about the university so that you understand. And Putra is named after our first Prime Minister, Tengku Abdul Rahman Putra. And that's the name that comes, Putra, to replace Pertanian. Yeah, so that it will remain, the name remain as UPM. And UPM is a research university, and we are also being awarded as the self-accrediting university in 2010, where we being self-accrediting, we can come up with our own program very quickly and doesn't have to take a long time to get the approval from the Ministry of Higher Education. So with the research university, there are 5 research universities in Malaysia. UPM is one of them. We have many disciplines, as I said, which include also a medical program, music, some other programs that are not agriculture related. We are placed in Putrajaya, and close to Putrajaya, within the multimedia super corridor, which is about only 10 minutes drive to Putrajaya. Putrajaya is our administrative capital, Malaysian the administrative capital for Malaysia, and it is about 30 minutes drive to Kuala Lumpur city centre. In fact, we are actually just across the road is already Putrajaya. Okay, let's go to viruses. All of you, I'm sure, have had classes on virus. Prof. Yola said that you have gone through. Just a quick revision that the virus very important because the virus can only replicate inside host cells. It is very small. It is obligate intracellular parasites and submicroscopic, which if you look at the size comparing the bacterium Staphylococcus aureus and some of the viruses there, you can see that the virus actually are very small. And the largest known virus in general is a mini virus. And Mycoplasma, you know that Mycoplasma is the smallest in between bacteria and virus. It is that if you want to compare the size of the virus, okay. And how small is the virus? When you say it is very small, how small is the virus? So that this picture gives you some indication of how small is the virus. If you compare here, what I put up here is compare viruses that cause diseases in poultry. My expertise actually in poultry diseases, specialized in avian medicine. So examples that I'm giving, most of the example during my talk today will be some that is related to poultry because we work on poultry diseases. So if you look at the viruses as compared to how small it is, you can see, the size of the tennis ball, 10 to the 8 size as if you compare to the others. And here, the biggest, the largest virus is a fowlpox virus. And the smallest virus in the avian group is the chicken anemia virus. It is a sickle virus. And viruses are unique. I think for those who work with viruses, you love the viruses, the work that we do. The viruses is cellular, and particles, virus particles, virions, as I mentioned earlier, do not grow or divide. And this is because the virus lack the genetic information that encodes tools necessary for generation of metabolic energy or protein synthesis or ribosome. So that's that's a spelling, not a spelling addition there. It's just the metabolic energy or protein synthesis, and they rely on their host to replicate or to multiply. Okay, the virus cannot multiply on its own. It depends. It requires source. So whether it is whatever host that the virus are in, the virus require that. If they are outside the host for some time, the virus will die because the virus cannot replicate or apply and multiply on its own. And the virus also using the host cell's machinery generates copies of its genomes to produce new viruses. So genomes is very important, and virus replication process varies on the virus genomes. Just now, Professor Zunita also mentioned about the genomes. They're similar, okay? They are the same in the context of genome. What are the importance of that? It's just similar to virus, to bacteria. And the virus infected cell is more like a factory because it will multiply within the cell itself. It's not like womb. Womb, you have one cell, then it will expand, become bigger and bigger, and not the same with virus. For classification of virus, I'm sure you're very familiar also, but I just want to highlight about the DNA and RNA. Virus, according to Baltimore classification system, actually there are several ways of how viruses are being classified, but the common one is the Baltimore classification system. Under this system, there are 7 classes. Class 1 is the double-stranded DNA, and class 2 is single-stranded DNA. So a lot we're talking about DNA and RNA. And The class 3 is double-stranded RNA, class 4 is single-stranded RNA, and class 5 is single-stranded RNA negative sense. 6 is the single-stranded positive sense RNA with DNA intermediate, and double-stranded DNA with RNA intermediate. I think if you forget about your viruses, your virology class, you can go back and revise about the viruses, because it is important to know the various classes. Then molecular biology, yeah, you talk about molecular bacteriology. So it's the same with molecular biology. Molecular biology is the study of the viruses at the molecular level. So we go into the molecular level. It's not just the whole virus, but go within and inside the virus to see the molecular at the molecular level. Because there are many genes, many genomes, RNA and DNA that we have to look into. And viruses also are very important for gene therapy and vaccination. Thus, the study of molecular level is very necessary. So when there are treatment, for example, using genes, various gene therapy, especially for cancer diseases. Now there are a lot of interest in using the virus for the treatment of cancer, including animal viruses for human cancer, but including animal viruses. And this is using the gene therapy. For in UPN, for example, we're also working on using Newcastle disease virus for the prevention or treatment of cancer. Okay, we have done several work on breast cancer, on colon cancer. So a few cancer, a group of researchers in UPM are working on it using various animal viruses. One of them is Newcastle disease virus. And also for vaccination, some of the vaccine need to use a molecular aspect. So that's why the study of molecular virology is very important. And in terms of molecular biology, if you talk about what is actually a molecular biology, this all deals with DNA, RNA, and protein. Okay, the molecular biology is an explanation of the flow of genetic information within a biological system, which states that DNA contains instructions for making protein. The DNA contains instructions for making protein, and these are copied by RNA. Then RNA is the one that uses these instructions to make a protein. So those are the important aspects of the molecular biology, whether it is in bacteria or whether it is in virus. So viruses, there are envelope viruses, and there are viruses that do not have envelope. Okay, so if the virus have envelope, then you have the envelope, then you have the protein, the viral structure there, the nucleus, then what is there in the virus aspect. And the nucleocapsid is very important also, the viral genome and nucleocapsid very important, and also the viral tegument. And Prof. Zulita also mentioned about genotype, phenotype, okay? And the many aspects of the thing that you see here, if you see that DNA, from DNA there is a transcriptional regulation, that means DNA transcribed into RNA, the various processing. You have the splicing aspect of mRNA or messenger RNA and translation before it is formed into protein. So that is the sequence of the things about what is happening in the virus. And various aspect of genome consists of either DNA or RNA, and genome is the one that will control our phenotype, and the genome is unique to each individual virus. Okay, that's why various virus will have either RNA or DNA, and they are unique that particular virus. Transcriptomics, this is again another way of looking at how the RNA transcript to further form in the later part to form the protein. Okay, so a lot of work has been done in terms of the transcriptomics as well as proteomics. Actually, a lot of research is ongoing with the proteomics, and Prof. Zunita also mentioned something about proteomics just now. And proteomics actually is the field that bridges the gap between genome sequence and cellular behavior. Some other when used for the diagnosis, for example, there are many conventional techniques. There are many gold standards. Then there are many ways of doing to extract and to detect a specific target or sequence or antibody probe. For example, for detection of virus, okay, for detection of virus, Newcastle disease virus, for example, the gold standard is to isolate the virus. That is the best way to get it confirmation that if you are able to isolate the virus. But sometimes there are viruses that we are not able to detect. Yeah, they are not able to detect, and we need to go further, and we need to use a molecular method in order to detect the virus. Dr. Prof. Zunita also mentioned about it just now. So the genomic mainly dealing with DNA, transcriptomics, RNA, then proteomics is on the protein aspect. These are some of the conventional method using the nucleic acid, either using the nucleic acid in the solution, and also all nucleic acids that bound to nitrocellulose or nylon membrane. So I'm not going to explain about the methods of doing all this here, because it will take long time, and I'm sure many of you have done it, and you have gone through the classes. And when you go through the classes about how the methods have been done, you'll know more about this method. So I'm not giving you one by one in terms of the methods that's been done for each of the technique that I mentioned afterwards. Prof. Zunita mentioned a long, I spent a lot of time talking to you about PCR, so I'm not going to go into this again. Just remember that PCR is an in vitro technique to generate large quantities of a specified DNA. And especially when you have a small amount, we have a small amount of DNA in various specimens, in various samples. Sometimes the sample, the amount is too small, and we cannot detect it in an ordinary method, in the ordinary way, or you cannot do the virus, you cannot isolate all that. So PCR comes handy because it can multiply a lot of the DNA in large quantity for it to be detected. Yeah, otherwise it will not be detected, but by using the PCR, you can detect. So the main 3 steps to copy DNA in PCR, you understand, is to open up the DNA by doing denaturation, find the target, what is the target they want to do, and this is called annealing, and to fill in a complete copy, or it's called extension. So what is being mentioned here is the same. And another one is when you want to do recombinant virus, then RNA is another way. RNA virus is a lot easier to do the recombinant virus. This is the co-infection of cell by genetically distinct virus strains that can lead to the generation of recombinant viruses. So many of the, not many, a lot of work that's been carried out now is to come up with a vaccine, recombinant vaccine. And mainly those are associated with the RNA viruses. Newcastle disease is one example. The process occurs in both non-segmented as well as within a segment of a segmented virus, or also co-infection that can lead to recombinant of the virus. And co-infection of cell by genetically distinct strains of segmented virus can also generate different combinations of certain progeny. I think for the development of vaccine, many groups in the world are working on recombinant vaccines. Some have come and produced a good vaccine, and they have come up with a good vaccine, but not commercialized yet, but good results have been obtained. And this is one of the ways of coming up with a vaccine where conventional vaccine does not work. I have here a short video about cloning, just to give some idea for those who are not very sure, just to give some idea. So we just listen to the video for a while. I hope the sound.
First step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. Restriction enzymes used in cloning produce staggered cuts in specific sequences in the DNA, generating fragments with cohesive ends. Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme. The DNA fragments are then incorporated into plasmids. The type of plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme, DNA ligase, is used to form phosphodiester bonds. The next step is to incorporate the plasmids into bacterial host cells by transformation. Each cell contains a different segment of DNA from the original organism. Taken together, these cells represent a DNA library. The cells can now be plated out on an agar medium. The colony of cells containing the desired cloned gene can then be identified and isolated.
Okay, so that's how cloning is being done, just to give an idea. But I'm sure you have been taught about the cloning, and it is good to have some information about it. Again, for PCR, I will not go into it, just to mention. I can't remember whether Prof. Zunita mentioned about real-time PCR as compared to the PCR. The real-time PCR is similar to PCR, but with a few steps extra. And this allows PCR reaction to be visualized in real time as the reaction progresses. Okay, and the real-time PCR allows us to measure minute amounts of DNA sequences in a sample. So it is similar to PCR, but it has more advantages by using the real-time PCR. Another molecular method is the study of the protein. And this study of the protein is using the Western blot. And it is very important because it can detect a specific protein in cell or tissue or organ or body fluid. And this technique also depends on the reaction of antibody with the protein that is immobilized on a thin membrane. So Western blot is a study of the protein, okay, to detect specific protein. The methods you can read, you can read or you can learn in your biology class later on. I do not want to, again, I do not want to go into detail into that. Just to give you an example of some information about Western blot. This is from the YouTube. You also can get this information later on. And the example given here is to Western blot for HIV. But they give you the information, some information about the principle of it. Sorry.
Today, we will be talking about HIV testing. We will be talking about the Western blot test. Okay, fine, whatever. The Western blot test is a multi-step process. It's a highly specific test used for HIV detection after preliminary examinations. The test rules out possible false positive HIV diagnoses. Western blot is a process that detects specific proteins in a sample, usually from antibody samples. In the case of HIV, the test detects the HIV proteins from the virus's envelope and core. The proteins are GAG, ENV, and POL. The antigen samples and proteins are added to a separation gel. Next, the proteins are transferred into a blotting tank with cellulose acetate sheets. Electricity flows through the tank to blot the sheets with the protein. The proteins blot the sheets of cellulose acetate because each protein has a different pH value where it ionizes. So each protein travels further along the cellulose acetate sheet before it ionizes. This is called cellulose acetate electrophoresis. Afterwards, labeled antibodies are added to the immune staining of blot. After that, testers perform autoradiography. Images on an X-ray film or nuclear emulsion are created by the pattern of decay emissions. Finally, antigen bands are visualized from the autoradiography. From there, comparisons are made between amino acid sequences to determine if the proteins attached to antibodies match those proteins found in HIV.
Okay, that gives you some idea about the Western blotting. Another common method also to use the flow cytometry, which is for immunophenotyping. We done some work. Some of our group have done it using for IBD, infectious basal disease virus. This is the last YouTube or video that I want to show you about flow cytometry, just to give you some idea again to this about what flow cytometry is all about.
Flow cytometry is a powerful technique for the analysis of multiple parameters of individual cells within heterogeneous populations. Flow cytometers are used in a range of applications, from immunophenotyping to ploidy analysis to cell counting and GFP expression analysis. The flow cytometer performs this analysis by passing thousands of cells per second through a laser beam and capturing the light that emerges from each cell as it passes through. The data gathered can be analyzed statistically by flow cytometry software to report cellular characteristics such as size, complexity, phenotype, and health. In this tutorial, we will look at how a flow cytometer works, how scattered light and fluorescence are detected by a flow cytometer, and how the resulting data can be analyzed. This view shows the primary systems of the flow cytometer schematically. These are the fluidic system, which presents samples to the interrogation point and takes away the waste. The lasers, which are the light source for scatter and fluorescence. The optics, which gather and direct the light. The detectors, which receive the light. And the electronics and the peripheral computer system, which convert the signals from the detectors into digital data and perform the necessary analyses. The interrogation point is the heart of the system. This is where the laser and the sample intersect, and the optics collect the resulting scatter and fluorescence. First, let's talk about how the sample is delivered to the laser. Here we see how the sample is transported through the interrogation point. For accurate data collection, it is important that particles or cells are passed through the laser beam one at a time.
Okay, I'm not going to show all. Sorry. Okay. I just want to show you, to let you know that there are many methods of these molecular methods which you can learn through YouTube. You can learn through YouTube. So if you go to various methods that you're not very sure, I mean, if you do not have laboratory classes, I'm not sure in Surabaya, in AIRLANGGA. But in Malaysia, students are not allowed to come onto campus yet. Our classes are being done online through Zoom or various other methods. Even the practical classes are being done also through Zoom. What we do is, if it is clinical, for example, the clinicians will videotape first about the techniques of doing things, and then show it to the student in class and discuss it in class. So what I'm trying to tell you here, that there are many of these methods which you can get through YouTube. So don't be despair if you're not in class, and you can see and you can watch over and over again. Immunofluorescence antibody test. This is another common test, blocking and immunostaining, that being used quite often in some of the detection method. And so with the molecular biology now, we are no longer just looking at individual genes. We have to look at many aspects of it. Again, I would like to stress that the genomics, transcriptomics and proteomics are the important areas now, or important disciplines that people are learning, working on it. So I think as students also you need to know more about genomics, transcriptomics and also proteomics. Yeah? And these are the things to summarize the thing, genomic, I mentioned that earlier just now. And we also have microarray, next generation sequencing are the new things that are important as being done. This is mainly to detect the specific target by using sequence or antibody or also probe, using the extract from DNA, RNA or the protein, and to examine the whole genome using the various methods. Microarrays, I think, is becoming very popular. Mass spectrometry also quite popular. So for microarray, it is also commonly known as DNA chip or biochip, and it is a collection of microscopic DNA spots attached to a solid surface. Also, many of this work for the viral disease, a lot of the tests are being used, microarrays being used as well. It's like that this microarrays is like to measure the expression levels of large number of genes simultaneously, or to genotype multiple regions of a genome. So microarray is being used. Okay, when you talk about the viral disease and why the molecular virology is very important, why we need to go at the molecular level, because one of the things that we mentioned earlier, to detect virus is not easy. Some of the viruses you will not be able to grow. You can only detect either through electron microscope or some other methods. And nowadays, the common methods used are the molecular level. And another aspect is that the virus tend to mutate constantly. Okay, it become very strange and some of them become more virulent. The range, the host, you can extended host range, vaccine or drug escape the mutants. So the variant strains, there are, especially when you talk about avian influenza. As I said, my examples are mainly the avian example. Avian influenza. It tends to mutate, and you get various different strains. There are other strains that come in. Coronavirus is another one. You're talking about the COVID-19. Now COVID-19, there are many strains of coronavirus, and there are also variant strains. And we have also found variant strain for infectious bacillus disease. So when you have variant strains, in terms of the protection, in terms of coming up with a vaccine, most of the time the common vaccine that we have will not work for the variant strain. So you have to look into what are the other aspects in order to come up with a good vaccine against the variant strain. And mainly we are looking at the molecular level, okay, by looking at various strains and serotypes to identify which is the area that we can come up, that are common genes together, so that we can come up with a vaccine that can target all those various strains. And when they mutate, most of the time they become more virulent, and the host range also extended. For example, some of the viruses that were not susceptible to other species before become susceptible to different species. And again, the good example is avian influenza. Before, avian influenza was not a problem in human, but because of some of the changes that occurred, the host range has already extended to the human as well. The disease spectrum is another one. You may have the same disease, but the clinical signs can be very mild, or it can be very acute, or it can be very persistent, and or some of them cause latent infection and immunosuppression. And that's make it very difficult to control the viral disease. And for disease management, you're talking, this I'm giving an example for poultry. but for some other animals, they may not be, the density, the stocking density may not be high, but for poultry, the stocking density is really very high. In some farm, you may have hundred thousands of birds in one farm, also in one shed that have been divided into various sections. Therefore, it is difficult to diagnose, especially subclinical and persistent infection and poor vaccine-induced immunity. So diagnosis of the disease, the common one requires confirmation. For the definite diagnosis, it requires confirmation based on the isolation of the organism. That's why I mentioned earlier that the gold standard for detection or for diagnosis of Newcastle disease is actually by isolation of the virus. In fact, for some other viruses as well. But sometimes it is difficult to isolate due to many reasons. Yes, and that we need to go to use the molecular level. Serology assays is one of them, and the most common one again when it is difficult to do the isolation is to do molecular assays, PCR and sequencing. Those are the popular one which Prof. Zunita has mentioned earlier. So PCR and sequencing is one of the method that is important in molecular biology. And also there is a need to differentiate between infected and vaccinated animals in order to eradicate the disease. And also very important sometimes when you're talking about zoonotic disease. This is what Prof. Zunita mentioned in her presentation. Actually, Prof. Zunita, I borrowed from your slide. Just to highlight again that molecular diagnosis is most appropriate if the pathogens are difficult to detect and identify. Because if it is easy to identify, then we will not go into the molecular level. It is easier to do the conventional method, because conventional method is usually cheaper than the molecular method. If you have to do the molecular method, you have to be sure that it is cheaper, faster than the conventional method. So molecular diagnosis is usually used for pathogens that are difficult to detect and identify, or it is highly infectious, dangerous to culture, or some that it's very difficult to culture. So then you go into the molecular diagnostic. Serology-based diagnostic, this is by detecting antigen or antibody, and it's very important that the performance must be sensitive and specific, cost and applicability. That also Prof. Zunita has mentioned just now. For any diagnostic test, you have to look into the cost and whether it is specific, sensitivity, efficient or not, then whether it is easily applied. Some of the common example, I think you are used to this also using ELISA. And if it is not use ELISA, because ELISA is one of the fastest method that you can do to test for numerous diseases. The non-ELISA methods such as HAIT, HA, hematoimmutation, hema inhibition, immunofluorescent antibody test, and neutralization test. And the use of monoclonal antibody based assay that able to detect specific subtypes. So there are also, it's not just looking into the particular disease, but also is able to detect the specific subtypes, and this we use monoclonal antibody. For diagnostic tests, the turnaround time is important. Again, this is very similar to what Prof. Zunita has mentioned. If you can do field or on-site testing, that is the best. You do need to bring the sample back. If you can come up with a rapid diagnostic test that you can do the test in the field itself, I think that is very good, especially when talking about large animal. That sometimes you are not able to. You want to do it in the field. You want to do it quickly, and if we can come up with the diagnostic test that has this characteristic, that'd be very good. So the turnaround time is very important. For Zunita also have already mentioned. If it take a long time to do the test, then the animal will have died by the time that we come up with the diagnosis. The cost per test, and especially if we have large number of sample that you need to test, the cost is very important to know. Whether multiplexing, that means many at one time, specificity and sensitivity. Some of the example, again, I want to highlight that ELISA, PCR, LANG, NASPA, microarray, all these are for detection of antigen, antibody, RA or DNA. Again, I will not want to go into PCR again, Zuneta has mentioned. So there are just to revise that nested PCR, multiplex PCR, in situ PCR. Okay, nested is very sensitive but prone to contamination, and multiplex, from Zuneta has already mentioned that it detects more than one target. So that's why you need to have more primers, not just one primer, and in situ is on the tissue section itself. The other different detection method include conventional agarose gel, real-time PCR, again more rapid and sensitive and able to quantify virus title by shedding itself. Well, I will just skip the PCR part, okay, because Zuneta has already mentioned. And for animals, whether it is whatever species it is, when come to viral disease, what is important, what is necessary is to prevent the disease. Most of the time there's no treatment for viral disease. So we need prevention, and prevention is by using the vaccine. And so that's why various kind of vaccine has been developed and come up to make sure that these animals can be protected against that disease. And if the virus structure change, or if there is mutation, or if there is a variant strain of virus that come up, new vaccine has to be developed. If they are not efficient or they are not effective for the new virus that has come up. So the ideal vaccine usually that should produce the same immune protection, which usually follows natural infection but without causing disease. Okay, what it means is that if the birds or if the animal is infected, usually they have some antibody that's produced, and if they can be protected, they don't die or they don't cause a lot of sickness before that, the antibody is the best antibody. So the immune protection should be the one that will produce high of the good antibody level, but without causing the disease. Then the vaccine must not cause the disease. And it should be a long-lasting immunity, and also interrupt the spread of infection. And again, the aim of vaccination, we know that to sterilizing the immunity, that means to prevent the infection, to prevent the disease. Sometimes the infection is there, but you don't see, if you can prevent the clinical signs, if you can prevent the mortality, that would be also good enough without the animals will not cause any problem or any side effects due to the organism. Blocking immunity prevent virus shedding and transmission of disease. And sometimes when you do the vaccination, the animal will be protected, but there'll be shedding of virus. And this shedding of virus, if it goes to other animals, they're not protected, then it causes disease in animal. So we must make sure that the vaccination will also cause blocking, will also prevent the virus shedding and also transmission of the disease. In poultry vaccine technology, just giving you an example, there are a lot of vaccine that's been produced. These are the conventional method. The live attenuated is other live virus, usually attenuated, or kill vaccine in different combination. It can be monovalent vaccine, that means for one particular disease, or one serotype, bivalent, trivalent, or polyvalent. And various kill vaccine also has been used using the adjuvants. And usually this kill vaccine or live vaccine, they are whole virus. Okay, we're talking about whole virus, not section of the virus, not part of the virus, not the molecular level. They are whole virus. But now, a lot of work has been, I mentioned earlier that recombinant vaccines are now for some of the diseases. Recombinant vaccines are already available commercially. Not many, but some are already available. For example, the viral vector vaccine, the Fowlpox virus, herpesvirus, and adenovirus, these are already available commercially. Reverse genetic vaccine, primarily for RNA viruses such as AIV and NDV. I'm not sure whether it is available in this part of the world, but some of those are already available that people have already used of that. So the knowledge on molecular virology is very important in order to come up with these vaccines. Again, some commercial recombinant vaccine, the example for fowlpox, we have for HPT, ILT, fowl adenovirus, the reverse genetic virus for influenza, ND and IB, against virulent RNA virus. These are the common one. And live recombinant bacteria for salmonella and transgenic plant. Some, the one with the asterisk, those are in the pipeline. It is not available in commercially yet. Yeah, the one, for example, for adenovirus, reverse genetic virus for NDV and IBV. Experiments, results, many have been published, but as far as the commercial production or commercial production, I may not be aware if there is a latest that is already in the market, but at the time that we were looking into various aspects, they were not ready for market. The proof of the vaccine is already there, but not available at the market at that time. Yeah, it may be that a company have already produced some of those. I've not checked up with that. And many genetically engineered recombinant vaccine, the viral vector vaccines, bacteria vector vaccines also are already available. Subunit or synthetic vaccine, DNA vaccine, and also reverse genetic vaccine, as I mentioned earlier. Okay, DNA. These are the viruses. So we have DNA virus and RNA virus. You know that for viruses, DNA is either DNA or RNA. We do not have virus that contain both DNA and RNA. And for example of the DNA virus, the common one, Marek's disease, infectious laryngitis, fowlpox, adenovirus. For the RNA viruses, these are for common important diseases. We have influenza, Newcastle disease, infectious bronchitis, infectious basal disease, lymphoid leukosis, reovirus, pneumovirus. Yeah, I just want to give some example of the work that we have done or some other group have done in terms of control of these diseases, avian influenza, Newcastle, IB, infectious bronchitis virus. They are RNA viruses, and Marek's disease virus is the DNA virus. So we look into avian influenza virus. These are the molecular aspect that we need to know about the virus. They are segmented negative sense single strand RNA genome. Remember the class that we mentioned, the Baltimore class of virus now and earlier on. And there are many strains of this virus, so that makes it very difficult in terms of the control of the disease. Hemagglutination, neuraminidase, acid, and then we have various protein, NP protein, M1 protein, M2 protein. All these various aspects of the protein, they are available that need to be targeted, and depending on the type of hemagglutination and also the combination of hemagglutination and neuraminidase, we have the various strains. And as of it is now, there are 144 avian influenza virus strains. Then in order to come up with a vaccine, we have to study all this structure to find out which virus they are similar and where we can put together so that a vaccine can be produced. If you look on this side, it's a comparison between human influenza virus and avian influenza virus. So you see that there are similarities in some of them, so that's why the avian influenza virus can also cause a problem in human. The new influenza virus isolated from bats is H17 and N10. So this we are not sure. Okay, these are from animals. The influenza virus that they are selected from bats. They may later on cause problem in human. They may later on. As it is now, we had, for example, we had bats virus from bats, the Nipah virus that Malaysia had some time ago, and some of the countries, Bangladesh and a few other countries that were affected with the Nipah virus, and these viruses were isolated from bats. And now we have isolated, and the researchers have isolated a new influenza virus from bats. And we have to be alert and aware of this because it may be another virus that can cause big problem in human. And again, very important to study the molecular aspect of the virus. For HA and hematopoiesis and neuraminidase, these are the immunogenic surface glycoproteins, and these are the very important protein if you want to do the differentiation or if you want to check into the various strain of viruses. These two, HA and A, are the important component. Okay, for influenza virus, you can see that the host range, there are many host range there, and all this may cross over to human, and somehow they cross over to human, and that's why we need to know more about the virus. Yeah. Type influenza virus. Okay, just some photos to show you the H5N1 in dogs and feline. Some time ago, I'm sure you have gone through that. There are many references. If you go through all these references, you can see what has been spoken about that. And comparing between H5N1, H7 and 9 infection in poultry, there seems to be more widespread and persistent, constituting a greater threat for human. Okay, these are some of the strains of the avian influenza in migratory birds, and how it gets into some other animals, and how it is spread into the animals and then get into human. And there are pathogenic infection. The H5N1 is H5N1 is mainly the highly pathogenic infection, avian influenza infection, whereas the H7 and 9, which was reported in 2013, they belong to the low pathogenic infection. Okay, just to show you how things happen with from the bird adaptation, mutation, and there is a reassortment that happened. H1 and 2 in 1957, reassortment happened. They get bird to human. In 1968, the H3N2 from the Hong Kong flu. Again, this is because of the reassortment that you have again bird to human, and re-emergence of other H1N1 strain, and the latest that cause problem. That a big problem is the reassortment that involve bird, pig, and human. Okay, other things may happen into that. So you can read some the papers in Trends in Biology, the example that I give here. If you go to this paper, you can have more information about this. Okay, just to quickly. Then for the detection, okay, detection of flock, unvaccinated flock, you want to detect whether what is the virus level, and in terms of detection and so on, the various methods are again being used. RT-PCR virus isolation is the gold standard that's being used. Again, for the detection of influenza, for diagnosis of influenza, the work that we do in EPM, we have come up with a rapid diagnostic test for avian influenza. And in fact, our rapid diagnostic test that we developed for avian influenza was the one that one of the methods that we use in detecting the avian influenza, the first avian influenza outbreak that we had in Malaysia. So our students and staff actually work in shift 24 hours to process the sample and to see whether avian influenza was there in that sample or not. And this is the method that we use again is RT-PCR. okay, real-time PCR. And we use various specific subtype specific primers and variation based on the PCR that what we have discovered. We the primers again, the primers we outsource, the primers that we outsource we do not do ourselves. And so besides the virus isolation and identification, we use also sequencing using the Sengus method and various other aspects. I just want to show you that our work, a lot of our work actually in terms of the virus detection is by using a PCR. And the real-time PCR that you use for the type A and subtype specific things that we use. Again, for the primers, we outsource those primers. Okay, we also use cyber green dye for simultaneous detection and subtyping of H9 and N2. And as mentioned earlier, that real-time PCR is more sensitive than virus isolation. Yeah, that's definitely PCR is more sensitive, especially it is difficult to isolate the virus. So these are the example again. Prof Zunita also have shown how to read those the bands. Yeah, and this is what we have done in the work that in our research. Internal control, again, we use a simultaneous detection, use cybergreen, cybergreen 1, based on the real-time PCR cybergreen for simultaneous detection and subtyping of H5N1, sequencing of AIV to detect subtypes. This will use many of these sequencing methods to detect the subtypes for highly pathogenic avian influenza. HPAI is highly pathogenic avian influenza, and low pathogenic avian influenza, and also the zoonotic strains by looking into the HA gene, the cleavage and NA gene, and neuraminidase at 20 codon deletion, and various aspect. Okay, the various aspect M2, NS gene, and also PV2 gene. I don't want to go into detail in this. So at the end stage, that it is important to control. Besides the biosecurity and vaccination, our ultimate goal is to prevent the introduction of AI. So this is very important if you want to to have the total elimination of avian influenza. As it is now, I think there are many countries that still have avian influenza. There are still some under control, some not under control, and we have to be on the lookout all the time because vaccination alone will not achieve eradication if you do not prevent the infection from coming in or from becoming endemic. Example again about the vaccine that are available, commercial vaccines that are available. Some of the countries are using this reverse genetic and kill vaccine. Malaysia, we have not. The government, the Department of Veterinary Services, have not approved using of vaccine. But I think Indonesia have already approved a long time ago of using the vaccine. I'm not sure whether this recombinant vaccine or reverse genetic vaccines are available in Indonesia or not. Okay, that just give you some of the information about the vaccination. If using the vaccination is very expensive, so that's why we need to find vaccine that are available that can be used easily in terms of the route of vaccination, to be easily and to make sure that the cross protection is very important, because we know that influenza there are different subtypes, yeah, and these subtypes, they have poor protection, cross protection. So we have to find and look into, work is still ongoing, to look into vaccine that can give a good protection for different subtypes. And for food protection, it requires hemagglutination and neuraminidase proteins. So these proteins are very important to identify, to look, and to see which are the common ones that can be used as a vaccine for the virus, so that it can have full protection, and so that the cross protection will be good. Okay. So right, I will skip some of this just to show again a lot of work is when come even for oral vaccination. Recombinant vaccine is the one that people are working on. All right, for as far as I know, no DNA vaccine has so far been licensed in poultry. In other species of animal, 4 DNA vaccine have been approved for veterinary use in some of the countries against West Nile virus in horses, infectious hematopoietic necrosis in salmon, melanoma in canine, and GHRH in swine. So so far in poultry, not that I know of, no DNA vaccine that has been licensed to poultry. So when you look into this, you'll see that how important that molecular virology is very important for the protection of animals, to come up with the vaccine and also to come up with the diagnostic methods. However, the DNA vaccine, there are limitations to DNA vaccine. It may be some of those may have inconsistent responses. A lot of work were in fact in UPM itself, our research group has been working on DNA vaccine as well. There is some response, but not as good as the response of using other vaccine. But still the work is still ongoing because there is the potential that DNA vaccine can be good, because it can be, require a large scale dose that should be cost effective and it should be easy to administer. Oral recombinant DNA vaccine poultry, also a lot of work is ongoing for this, especially using the attenuated salmonella as the delivery method for H5 DNA vaccine, and some other methods using silver nanoparticles and encapsulated H5 DNA vaccine. One of the students have been working on the silver nanoparticle and encapsulated H5 vaccine. We are still working on it. There's some showing positive result, but a lot of work still need to be done. Okay, that's about the H5 avian influenza. I want to go next to for the way forward for control of avian influenza is to have diagnostic tools that are sensitive for rapid on-site detection. And this again, work is ongoing. We have come up with a diagnostic method, but we need to improve on that as well so that it can be done and it can be quickly done on site. The work on site is very important, that we do not have to bring sample back to the lab. Okay, Newcastle disease virus. These are the structure of Newcastle disease virus. Again, the most important one is the HN protein and also the F protein. And okay, the peptides that will give you some review class on the Newcastle disease. I will not go into this, but just to show you some of the important aspect when we want to identify the viruses, which group that they belong to, whether they are virulent and which type, and also to compare with other countries. The class 1 virus, we found that the class 1 virus probably has a worldwide distribution in wild birds and often isolated in live market. The various genotypes. Okay, I'm showing this just to relate to you the importance of molecular virology in prevention and also in the detection and prevention of diseases. And from the work using the molecular aspect, we were able to discover, we were able to genotype this virus into the various group. And by genotyping them in various group, we're able to see in terms of the vaccine that have been developed, whether those vaccines that have been developed are sufficient to control the various genotype. For example, the genotype 7 that have been discovered. This is the latest genotype for Newcastle disease virus. we found that the vaccine that are available commercially now, they are not as good in terms of the protection for genotype 7 virus. Though the birds are vaccinated, but the bird can still come down with the disease. This is the complete genome sequence of several NDV genotype 7. If you see here, okay, there are a lot of work that involve molecular virology. Okay, again, just comparison that we have isolated several isolates of genotype 7 NDV genotype 7 in Malaysia. So a lot of work has been done in terms of looking into the diverse aspect of the isolates and how to compare them with the Lacerta strain. Lacerta, you know that it is one of the oldest strain that's being used for the prevention of disease, for Newcastle disease. Again, IB, virus strain. I will stop in 5 minutes so that we have time for question. All this I'm showing just to show that the example of how when various strains come in and what we need to do the various aspect of detection. And IB infectious bronchitis virus is another virus that is difficult to control. It's difficult because the vaccine that you produce may not be effective for another strain that comes in later on. And IB also has a lot of mutations that comes along the line. So it is not like for Newcastle disease. Newcastle disease is easier in terms of coming up with the vaccine, but for IB, it's another one that's difficult to control. So working with the recombinant vaccine would be one of the ways in order to come up with a vaccine that's able to control infectious bronchitis virus in chicken. Yeah. Because the virus is very persistent and usually have a long-term shedding. So we need to find a vaccine that can reduce or can block the emergence of various strains and also the virus strains that may undergo reversion to virulent strain. So the vaccine, the vaccine that we produce should be able to be safe for the animals and to produce good antibody. Okay, this is in terms of the control. And the last one that I want to touch on is the Marek's disease. Marek's disease is a DNA virus. Again, there are many serotypes. There are 3 serotypes there. And the vaccination with the cell-free and cell-associated vaccine of the live attenuated serotype, the response is, and also HVT are the common one. Okay. Again, if you look at this, in order to come up with the vaccine, you have to study all this structure, all this, the molecular level, the glycoproteins, and looking into various aspects of the virus in order for us to understand better, okay, the genome wide sequence analysis, to do the genome wide sequence analysis to check for the several genes that involved in the process and in the work that we want to do for the treatment and also for the prevention of the disease. Marek's disease, we know that it is an oncogene. It causes a lot of problem, especially in earlier time. It was in young birds that we have this problem, and sometimes the disease comes up because of the genetic resistance that they come in, and sometimes the vaccine that has been used is no longer able to stop the virus from developing or evolve into another strain, a type of virus. Okay, the control, the challenges in controlling is that we have to identify and appreciate the virulent strain from the attenuated serotype and one vaccine. And early diagnosis is very important in the control of the disease. Okay, I will not go into this, but very important to know that pathogens are moving targets. So a quick way of detecting the pathogen is by looking into the molecular methods. Right. I also would like to acknowledge some of the slides. Actually, I got it from Prof. Omar and Dr. Noyasmin. And as you notice, one of the slides that I borrowed from Prof. Zita. Yeah. So, Dr. Aan,
Yes, Prof.
I'm done.
Okay, thank you,
Okay, thank you very
Professor
much.
Aini. Thank you, Professor Aini, for the insightful presentation. So now we are going to take a question.
I hope the voice is okay.
Yeah, I think
your
connection,
mic is
Prof,
not good. Yeah.
It's unstable. Can you hear my voice?
Yeah. Prof, Dr. Ann, your voice not very clear.
Hello?
Yes.
Hello?
If there is question, I think it is good to put on the chat. If the question, it's good to put on the chat.
Can you listen me, Prof? Can you listen me?
Yeah.
Clearly?
Not clear. Is that Dr. Saiful? Dr. Saiful, can you write in the chat? I think it's better if you write in the chat.
Yeah.
Ya, untuk yang kelas yang reguler dan internasional, silakan dipersiapkan pertanyaan ya, bisa by chat, boleh bahasa Indonesia.
Ya, boleh. Mungkin tak ada soalan, Dr. Prof. Yola. Mereka sudah penat agaknya.
Excuse me, Prof. Hello.
Hello,
yes.
Yes.
Ya, nah,
I want to ask, Prof. Excuse me, before I tell about my asking, can you hear my voice clearly, Prof?
Yes, can hear, yes.
Yes. I read about subunit and recombination method for creating vaccine, Prof. In this method, is need a carrier virus that contain or brings an immunogenic antigen. Are there any requirement for choosing the carrier virus that will be used? Thank you, Prof. Brings immunogenic requirement for choosing the
requirement for what? What requirement that you ask?
and is there requirement
Kak,
for choosing the
Kak.
carrier virus that will
Kak.
be used?
Bram, kalian matiin dulu internetnya. Mimi nggak bisa nih internetnya putus-putus. Kalian
Lanan, I think Lanan, I prefer to you write in the chat.
Yes, I will, Prof. You bring other using the Prof.
There is one question by there is one question by Dr. Saiful, which asks which is the best sample for molecular identification of avian influenza. There are many samples that you can take. The best would be the respiratory from the lungs. From the lungs would be one of the best samples that you can take to use.
No.
Sorry, I cannot hear.
Text.
Yeah, I cannot hear. I'm sorry. Your voice breaks.
Ya, gangguan ini, Prof.
Prof Aini.
Usually we use swab sample, Dr. Saiful, the swab sample. Dr. Lanam, various virus have been used as carrier, and the easiest virus that we have worked so far is by using the Fowlpox virus. That a lot of work has been done by using the Fowlpox virus.
Yeah. Hello.
The question by Dr. Ratri. Sorry, can you, Zunita, can you hear my voice? Is it clear or is it breaking?
Yeah, it's very clear, Prof.
Yeah, okay. Because I'm hearing. It's clear, right?
Yeah, from your voice it's clear, but then from Uner is a bit like there's echo.
Yeah, that's why it's
echoing there.
breaking up on that side.
Voice is coming from there's
Dr. Saiful, I will answer your question later on. Can I answer because yeah.
Yes, Prof. Aini.
Yes.
Question in chat.
Yeah, it is not.
Yeah.
Yeah. Can I give that information to Dr. Saiful or to Prof. Yola afterwards so that it is easier?
Nggak dengar.
Ini
apa sinyalnya nggak bagus ya. Jadi
the question about the cost for PCR, Professor Zunita has already mentioned. It depends. It depends on what. This is PCR, right? Or COVID. If you want to know, if you want to test COVID with PCR, the cost how much? But for the cost, on the cost for the test include not only the materials but also the what they call the cost of the people that handle that as well.
Sorry.
Yeah. So that's why it depends. I mean, the cost varies. One is the material that's being used, and also the person that handling the samples. Yeah. On my side here, I can hear echo. Echo coming out.
Excuse me, Professor.
Yeah.
May I ask some question?
Yeah, sure.
Okay. My name is Ito. I have a question. As we know, to create a vaccine, sometimes we must test our vaccine to animal testing like rat or monkey or any more. But so my question is it possible that the virus that we inject inside our animal testing can mutate it or evolve, then create a new disease or a new virus or a new symptom? Thank you.
Okay. For virus for vaccination, usually we do not use in rats or in mice. Usually we do our tests direct into the host. So if the vaccine, if it is for poultry vaccine, the work is direct into the host, into chicken. We do not put into rats first or mice first before we put into the chicken. Because the animals are easy to get, even for if you have the vaccine for large animal or cattle, for example, we do not go into rats first. We straight away to the animal. Yeah. The possibility of creating up a new strain, I'm not sure because we have not done it in other species of animal. Yeah? We go direct into the host. It's the best way of doing the vaccine trial. You do direct into the host. We have produced quite a number of vaccine and for cattle, for goats, but these are being vaccinated directly into the animal. Our sample, our sample for trials are the host itself. So if it is for a cattle vaccine, then it's cattle. If it is for goat vaccine, we use goat. And if it is for chicken vaccine, we use chickens. Because that's the best way, yeah? The best way. If you put into other species of animal, the results that you get may not be the same as the results that you get when you put into the host
Bu, ini
himself.
Okay. Thank you, Professor, for the answer.
Okay.
Good afternoon.
Thank you. Good afternoon. Thank you.
Excuse me, Professor.
Yes, who's that?
I'm Grace. I'm from class of Banyuwangi. I want to ask you a question. May I?
Yes, please proceed. Yeah.
Can you hear my voice?
Yeah, I can hear, but it is breaking. I'll try to understand.
Pardon?
I will try to understand. Your voice breaks, you know, in between. Some I can hear, some not so clear. But you try first if I can understand your question.
Oh, okay. Here's my question for you, Professor. As we know that you recently taught us about molecular diagnostics that is most appropriate for pathogens that are difficult to detect and identify and highly infectious, dangerous to culture, and non-culturable pathogens.
Yes. Your question?
Tests. And my questions are, is there really any correlation between resistant virus and molecular diagnostics? And can virus that has resistant in it diagnose based on molecular diagnostics? Thank you, Professor.
The correlation between virus and molecular diagnostic. Was that your question?
Yes, and can virus that has resistant in it diagnose based on molecular diagnostics, Prof? Thank you.
You are talking about diagnostic. You are talking about diagnostic. So diagnostic, it does not matter whether it is resistant or not. Diagnostic is to identify the organism or the pathogen. Okay, so whether that pathogen is resistant to whatever it is, it does not matter because you are looking into the the pathogen itself. Yeah, are you looking at the molecular level or are you looking at the whole organism? So there is no correlation in terms of that. It is just that the virus, you have the specific target that you want to do. So if you are looking, for example, if you use PCR, what are the targets, and you have your primers, or you do sequencing, you'll be able to detect what you want to identify. You know, using the right primers, you should be able to detect.
Okay, thank you, Professor.
Okay, you're welcome. Any other question?
Ya, silakan untuk kelas yang reguler lain bisa bertanya pakai bahasa Indonesia, tidak apa-apa ya.
Tidak apa-apa ya.
Hancur Mimi.
Malu, Prof.
Malu ya. Tak apa.
Terus kalau maluin aja. Ya Allah,
Kalau malu
Tuhan.
tak apa, dia boleh tanya nanti.
Kok ya masalah kayak
jawab.
gini.
Excuse me, Prof.
Yes.
Good afternoon. My name is Siti. I
Yeah.
want to ask you question about molecular tracking. Nowadays, we can do molecular tracking by
Bisa, tetap kan memang kayak gitu.
Sorry?
Sorry, tak dengar. Tak. apa tak jelas. Saya kurang jelas soalannya.
Saya sudah ini mengetik di kolom chat pertanyaan saya.
Maybe Prof. Zunita boleh jawab soalan ni? Lebih soalan Rindi ya?
Mana?
Rindi, Rindi pasal
molecular docking.
Biochemical compound. That.
I'm not familiar, unfortunately, about this. Rindi.
Saya pun tak familiar yang orthodox bina ni, saya tak familiar. Soalan lain Rindi, saya minta maaf, saya tak familiar pasal ini tentang especially pasal biochemical compound. Saya bukan biochemist, saya jadi kurang jelas dekat situ. Dan teknik ni pun saya tak pernah guna. Saya tak dapat bagi jawapan lah situ.
Okay, Prof. Thank you very much.
Maaf ya Rindi.
Gangguan
ada lagi?
Deras hujan deras di rumahnya Prof. Aini because hujan deras.
Ya, itu kan tadi suaranya
hujan
okey juga, tapi sekarang ni
because rumahnya
tak boleh dengar
Prof. Aini, maybe it's time to closing remark.
Wah,
putus-putus juga.
Sorry, saya tadi silap hantar pada Saiful.
Bagaimana, tidak ada yang bertanya lagi? All students?
Tidak ada soalan lagi?
Excuse me, can I ask?
Yes.
I'm gonna ask regarding COVID-19. So why in COVID-19 infection the lymphocyte decreases in contrast with other viral infection and cancer as well?
Okey, saya COVID-19 ni saya tak begitu tengok betul-betul lah, jadi saya pun tak mau bagi jawapan. In case jawapan saya ni tak betul, sebab saya takut itu detail lah tentang COVID-19 ni. Saya tak mau nanti nak bagi jawapan in case jawapan ni tak betul itu. Saya rasa ada banyak yang ditulis tentang COVID-19. banyak artikel yang telah ditulis lah, tapi sebetulnya saya tak do the molecular level of COVID-19. Prof. Zulita kalau jawapan tentang COVID-19 ni.
Ya, gimana? Baik.
Okay, thank you.
Oke. Oke, kalau tidak ada pertanyaan.
Assalamualaikum. Suara saya bisa didengar tidak, Bu Yola?
Ya, dengar kan? Ya, dengar.
I'm sorry for the.
Silakan.
I'm sorry for the inconvenience because now. Hello.
Ya, dengar.
Oke, boleh dengar? Bisa didengar?
Boleh.
Okay. Due to the limited time, so can we end the sessions of question and answer and continue with the next agenda?
Ya, okey. Saya okey.
Saya pun okey.
Okay.
Terima kasih.
Okay. I'm sorry for the inconvenience because of the unstable of my connections. Okay. I would like to say thank you to our speaker for the informative and insightful presentations. So finally, from today's lecture, let me try to recapitulate the message from our two speakers. Okay, so from Professor Dr. Zunita, she has explained the diagnostic tools both on the conventional and molecular base as well. And we note that diagnostic tests are essential tools for confirming the health status of animals and for identifying pathogens. Therefore, detections, identification, and characterization is crucial for the best antimicrobial therapy. And from Professor Dr. Aini Idris, she has explained from the basic of viruses to the molecular level, and also about the diagnostic tools and the vaccine development for viral disease control. And we note that the way forward in the control of viral infectious diseases, especially in poultry, need to integrate the use of vaccines and diagnostic in management and biosecurity of the farms. Okay, once again, thank you, Professor Zunita and Professor Aini for the insightful presentation. So now we come to the next agenda, which is certificate awarding that will be given by Dr. Yola Rahmahani.
Oke, terima kasih Dr. Aan. On behalf of the Faculty of Veterinary Medicine, University AIRLANGGA, I would like to hand over the certificate of appreciation to our distinguished speakers. Virtually, our first certificate, will Dr. Zunita Zakaria, Professor at the Department of Veterinary Pathology and Microbiology, Pathology and Microbiology, Pathology and certificate. Asia, Duka Dr. Aini, SDM, Duka Dr. Liz, medical study, veterinary, and again, adopting or Idris, medical study, Dr. Idris, Dr. Clinic Faculty, Dr.
Okay, thank you, Dr. Yola. Now we come to the next agenda, which is closing remarks from Secretary of Microbiology Department, Dr. Wiwi Tiasningsi, Doctor of Veterinary Medicine, Master of Health. For Dr. Wiwi, the time is yours.
Okay. Thank you, Dr. Aan. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Everyone. Alhamdulillahirabbil alamin. For delivering the material in this guest lecture. Your lecture about molecular, there is a slight distraction, Prof. Yeah, sorry. Hopefully, your explanation. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Dr. Aan.
Dr. Aan.
Yes.
Dr. Aan.
Sorry.
Sudah.
Yes. Wiwi sudah menutup.
Yeah, I'm sorry.
Yeah,
thank you. Thank you, Dr. Wiwi, for the closing remark. So, finally, we get the last session in this event, that is closing. So, on behalf of the committee, we would like to thank to all the speakers for the informative and interesting presentation. And thanks to all the participants for the participation. May that we have conducted today will be useful for all of us. Once again, if there are any imperfections during the implementation of this event, we would like to apologize to everyone. So let's close this event by saying together. Alhamdulillahirabbil alamin. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you very much. Thank you, Professor Prof. Zunita, Prof. Aini,
Prof.
Thank
Bu.
Thank
you, Dr.
Thank you, Prof. Thank you, Doc.
Thank you. I'm sorry, ini problem because of the bad connection. I'm sorry.
Prof. Sunita.
Makasih, Prof.
Sekali lagi.
Bye.
Aduh.
Thank you.
Ini saya sampai keluar rumah cari jaringan.
Ada Prof. Rahayu, tadi belum
Prof. Rahayu.
Prof. Rahayu.
Belum sempat reunian ya, Prof.
Prof Aini, Prof Zunita, please send my best regards to the lecturers in the UPM.
Inshallah.
Inshallah.
Once again, thank you for accepting our invitation, become our speaker today in our event.
Yeah, my pleasure. Pleasure, yeah.
Yeah, thank you for the opportunity.
Yeah.
And
lagi bagus kalau ke sana.
and I would like to apologize because of the technical problem during this implementation for today.
No worries, it's okay.
Yeah, no problem.
Yeah.
Tahun depan kita ke sana, Prof Aini.
Ya, insyaAllah.
Kapan kita bisa ya, Prof Aini?
InsyaAllah, insyaAllah.
Ketemu.
It
Prof
will be
Zunita,
ditunggu Prof. Welcome you.
Ya, sayang pandemi. Kalau tidak ada pandemi, ini bisa Surabaya.
Ya,
ke Surabaya atau kita ke sana lagi.
Ya.
Most welcome.
Welcome, ya.
Okay, Alif.
Yeah, thank you. Thank you, Bro.
Thank you.
Thank you. Thank you very much.
Terima kasih Bu Wiwi.